É verdade, mas a gente às vezes baralha-se.
Good morning, everyone. Welcome to another event celebrating 30 years of Infarmed, advanced therapy medicines from innovation to patients. I call to the stage the President of Infarmed, Rui Santos and Miguel Forte, President-elect of the International Society for Cell and Gene Therapy, to begin the open session. Dr. Rui Santos has the floor.
Thank you, Wanda, for the introduction. Bom dia a todos. Vou dizer 2 ou 3 palavras em português, só para saudar todos os que estão aqui no auditório. Muito obrigado pela vossa presença. Good morning to all of you. I will now speak in English since this will be the language that we will use during this meeting. So I would like to welcome you all in this auditorium at Infarmed, and also welcome all those that are following us online, because this meeting is also transmitted online. So some people can also follow it through our channel in YouTube during the day. I would like to give a special welcome to Professor Miguel Forte, who is the President-elect of the International Society for Cell and Gene Therapy, and also Miguel is a good friend of us, not only of me, but also of us, because he had a very important collaboration with Infarmed some years ago, and we kept this very good relationship, and it's a pleasure that we can have this meeting organized in collaboration between Infarmed and also himself and the society. Allow me also to give a special welcome to Professor Bruce Levine, who will speak just after this introduction in this meeting. We are sorry that we cannot have Professor Levine here in the room, but unfortunately he was not able to come due to a circumstance, and I would like to thank him very much for still being in the meeting through the digital connection, knowing that being in the US is an effort for him to be at this time in Portugal, although we are in the West Coast, a little bit closer to the US time zone, but still an important difference. Of course, I would like to say a word of welcome of the different persons here. I know that they are from many associations that are involved with medicine, so the pharmaceutical industry, pharmacies, the societies, also universities, research center, hospitals, and of course very much also patient associations. So we have a broad audience, which I hope will contribute to making a very fruitful meeting. So again, welcome you all. And allow me now to also to say a few more words in this introduction. So in the year of 2023, Infarmed celebrates its 30th anniversary. It's a very important milestone in the life of our agency, and we have been celebrating it by organizing a number of events that not only portray our activities, but also look into the future of medicines and medical devices regulation. Today we commemorate innovation, because when we talk about advanced therapy medicines, we are talking about the most innovative field of medicines. In accordance with European legislation, and already in particular referring to Regulation 1394 from 2007, which deals with advanced medicinal products, as well as the establishment of the European Medicines Agency, and also other relevant regulations. So Portugal and Infarmed, through various stakeholders and external experts, have contributed to the development of ATMPs and also introducing these medicines into our market, some of them quite at early stages. This has been done through national and the EMA level scientific advice in the evaluation and approval of clinical trials with investigational advanced therapy medicinal products, and also through the participation of our representatives both in the Committee of Advanced Therapy Medicinal Products, CAT, and also the Committee for Medicinal Products of Human Use. And you have our colleagues here in the meeting. The number of advanced therapy medicines under development, evaluation, and authorized in the EU has grown impressively in recent years. These figures are actually a reflection of the innovation in the area of medicinal products for human use. Given the importance of innovation, the discussion around the development of these particular medicines is a crucial theme we also wanted to pay special attention during this year of our 30th anniversary. As such, and as I mentioned already, we are very honored to welcome Professor Bruce Levine from the University of Pennsylvania, who has been instrumental in the creation of CAR T cells as a solution for the treatment of certain cancers with very few therapeutic alternatives. I would also like to welcome Professor Luis Almeida from the Faculty of Pharmacy of the University of Coimbra and his research center, who through his research seeks to find alternatives with gene therapy. Today, our aim is to focus on the authorization of these particularly medicines, as well as to highlight and discuss regulatory challenges that authorities and developers are now facing. And I know that this will be well referred. Collaboration and knowledge sharing are the bedrock of advancement in this field. To this end, we will hear from Dr. Isabel Vieira, which is the alternate member of Portugal in the Committee of Advanced Therapies, and also Dr. Margarida Menezes Ferreira, who, as a former scientist of Infarmed, has contributed decisively to making ATMPs a reality at European level. Thank you, Margarida, for this. Through her participation in CAT and also in the Biologics Working Party as a Portuguese representative, and also today as an independent consultant in this area. So she's very active, and she was telling me that she will be actively speaking here, so we will await her points. In the quest for better health and improved patient outcomes, ATMPs have emerged as beacons of hope. These groundbreaking therapies, including gene therapies, cell therapies, and tissue-engineered therapies, represent a paradigm shift in how we approach and treat these diseases. The goal of these medicines is to reach those for whom may not have treatments or cures for their diseases, such as the genetic diseases. To further address ATMPs, the importance of ATMPs, I will also welcome Dr. Martina Schussler-Lenz from the Paul-Ehrlich Institute in Germany, and Dr. Schussler-Lenz is the former president of the Committee of Advanced Therapies, and she will address the regulatory perspective of ATMPs research and market along with patient access. So thank you also very much, Martina, for being with us today. She will be joined also by Dr. Felipe Moita from the Portuguese Oncology Institute in Lisbon, whose expertise focuses on the use of these medicines and treatment in Portugal. The genesis of these biopharmaceuticals truly lies in the academic institutions and small and medium-sized enterprises, which by virtue of their research and development endeavors paved the way for the initial strides toward prospective regulatory approval and utilization of these therapeutic agents. For this reason, we also have considered essential to discuss the historical narrative of criostaminal. This will be addressed by Dr. Francisco Santos, who is the Director of Production and Development at criostaminal, who is also in the development of ATMPs and in particular cell therapies, and therefore actively contributes to facilitate patient access to experimental advanced therapeutic medications within the country. It is widely recognized that these biopharmaceuticals represent the costliest medications authorized. This high cost is in some extent associated with development expenditures. Furthermore, a significant contributing factor is their designation for the treatment of rare diseases, a category that yields limited economic returns for the stakeholders who have invested in them. given their high pricing and often lack of robust clinical evidence of the added value, access to these medications poses a potential threat to the sustainability of also the national healthcare systems. Consequently, it becomes imperative to underline the challenges in assessing added therapeutic value and cost effectiveness of these technologies and how we can overcome these challenges. To address this matter, we will have the insights from my colleague, Professor Claudio Furtado, who is the Director of Health Technology Assessment here at Infarmed and also involved in the pricing and reimbursement issues that we also handle here at Infarmed. The primary objective of those involved in the development, in the assessment, in the authorization of medicines is, of course, the well-being of individuals. We shall incorporate then the insights of Dr. Mencia de Lemos Belmonte, who is representing the Spinal Muscular Atrophy Foundation. Dr. Belmonte is affiliated with patient organizations nominated by the European Commission to serve as member of the Committee of Advanced Therapies. And to conclude, allow me a final remark, also as President of Infarmed, also to signal some ongoing key developments that which also express the importance of today's meeting for this purpose. And I'm referring here to a number of legislative developments that are occurring in Europe, some showing also pioneering work at different stages, and this will be framing the future developments in the areas that we are also discussing today. On a more transversal perspective, I can mention two pieces. One is the European Health Data Space Regulation, which is very important, and the other one is the Artificial Intelligence Act that are about to be approved. So we expect that these two regulations will be approved quite soon if the ongoing negotiations will achieve its purpose. But besides these two pieces, which are more, as I said, more transversal in terms of its application, we have two other initiatives which are closer to the discussions that we are having today. One is the Health Technology Assessment Regulation, which was published already in 2021, and that will be applicable from January 2025. And this will be very impacting, very important when it comes to advanced therapies, because also this regulation, which will be implemented in a gradings in different stages, will start with oncology medicines and ATMPs, and of course they also overlap in terms of its application. But there will be also an important development, which is the reform of the pharmaceutical framework that will be further discussed from 2024 onwards. This is a proposal that the European Commission has presented in the last April, and it has not yet really started to being too much discussed at the level of the member states. This will start mainly with the Belgian presidency from January 2024, but you have seen already that the Parliament has already given quite a lot of attention to these proposals, with the two rapporteurs discussing and coming with different perspectives, with quite opposing options in terms of what they are proposing. Let's see what kind of compromise we'll be able to find in terms of finalizing this file, which will take still a few years. Of course, these activities are all focused in the patient. They are all aimed at taking the best solutions to them, which at the end of the day, it's our responsibility of our work. Therefore, I would like to conclude now. I'm sure that today's discussion will constitute a sound contribution for the future discussions also at your level, and it's also underlying the importance of the dialogue that we'll have to establish between regulators, developers, universities, research centers, patients, health professionals, and so on. I wish so to extend our sincere gratitude to all our esteemed guests for accepting our invitation to partake in today's event, dedicated to honoring the contributions of Infarmed to the field and for enhancing the programs through their valuable interventions. I would like to conclude with a special thanks both to Professor Miguel Forte of the International Society of Cell and Gene Therapy, and also my colleague, Professor Fátima Ventura, of the Scientific Evaluation Unit within the Medicines Evaluation Department at Infarmed, and also she serves has alternated in the CHMP committee of the EMA. So thanking both of them for coordinating the two sessions that will be addressing the aspects that I have tried to underline on my introduction. So thank you to all the participants, and I'm really looking forward to a very fruitful and insightful discussions during this morning. Thank you again very much for being here. Miguel.
Professor Miguel Forte will now take the floor.
Obrigado, Rui. Bom dia a todos. And now I'll switch to English. It is a huge pleasure to be here because it combines two things. One is the fantastic innovation, exciting science, challenging development that we're all involved in bringing value to patients, and doing that with friends. It's all about people, it's all about the efforts that we do, it's all about the vision that we have, and the intention that we all share of generating this value that will bring solutions to patients. And being in Portugal, being at Infarmed, and being with friends, it excites me. I will not take too long, but I just wanted to say that Bruce Levine, that will be talking in a minute, and I'll introduce him properly, once said that 10 years ago or 15 years ago, cell therapy was a cute thing. People are looking at us and say, oh, you're having a bit of fun, right? Today, it's a reality with multiple products in development, accelerating approvals, a significant business, and above all, bringing solutions, long-term solutions, nearly or actually curative solutions to patients. We all have a role in that development. We all have a role in that access to patients, and that's what we will be discussing here today. The challenges are significant in the current time. I was just chatting before coming in that we may be in a lull of activity, excitement, financing. There's some challenges also in terms of the number of clinical trials. It is the growing pains of a maturing field, and resilience and ability to continue to go forward, and above all, in partnership between all stakeholders. And that's what I think is represented here today by sitting at the auditorium of a regulatory agency that has been impacting and part of this development for a long time, bringing in organizations like ISCT that combines regulators, academics, and industry, and others that are interested in developing these fields with the right generation of evidence and the right access to patients, but also a lot of you in industry, in the organizations that take the risk, the investment, and the pleasure and the reward satisfaction of bringing value to patients in the end. That said, let's then go on to the discussions. And the first one is a presentation from Bruce Levine, another very good friend, who unfortunately had a silly accident. All accidents are silly anyway. Had a silly accident and could not travel, which I can tell you for him is unheard because he's always on the move. So I had the intention of taking him around and showing at least one of the things that for me is a highlight every time I come to Portugal, which is this restaurant on the Praia da Draga. And no publicity, but it's a fantastic restaurant. But unfortunately, he said we have to do it some other time. Bruce is the professor at the Barber and Edward Netta professor in cancer and gene therapy at the Perelman School of Medicine at University of Pennsylvania, UPenn. He was, together with Carl Jung, instrumental on the first steps. I think there was a watershed of change when we saw the initial CAR-T clinical results. And it is all about the clinical results that matter. That change created our field to what it is becoming now. He will take you through that. Another interesting point, which we also share, is that he started his career in immunology and infectious disease and HIV, like I did here in Portugal as well, and for some time. And that brings me an example, which is we saw HIV appearing, we managed it, and we've now turned it into a tool, the steps of the vectors that Louise will talk. And I always think that how we can really transform a challenge, an adversity, into an opportunity, into a tool, into something that delivers our desires. Bruce has more than 200 papers published, and he has another thing in common. He is the immediate past president of ISCT, so I follow on his path, and that is more than what I can say. I mean, I've been following, learning, and enjoying his leadership behind him. So, Bruce, thank you very much for doing it at a distance. Over to you.
Thank you, Miguel. Thank you for your very kind introduction. Thank you to the organizers for the invitation and opportunity to speak to you. I'll just ask for confirmation that I have my slide in presentation mode and that you can hear me okay. Okay. Hearing no protest, I will continue. These are my disclosures. I have a financial conflict that is managed in accordance with University of Pennsylvania policy and oversight. So why am I virtual? So that's the picture on the left. The correction is in the middle, and my left leg is locked at 0, prohibiting me from sharing in that exquisite restaurant advertised by Miguel. So I want to start by asking the question, how did we get here? How did we get here where our first adult patients are 13 years cancer-free, the first child in the world treated with CAR T-cell therapy is 11 years cancer-free, and in fact, a freshman at the University of Pennsylvania. And where are we going? So let's start first with how we overcome the scarcity of tumor-specific immunity. Now, cancer is derived from your own cells. The immune system did not evolve to recognize cancer because it is seen as self. Nevertheless, due to some mutations or virally infected cells, there are some rare T-cell receptors that do recognize cancer, and those can be isolated and delivered genetically. But the challenge is the HLA type. So each T-cell receptor only recognizes a certain percentage of the population. And you can see in Europe, even with 5 HLA types, you're not covering 100% of the population. Now, CARs recognize surface antigen, and they're MHC independent, composed of an antibody binding domain and T-cell signaling domains on the intracellular surface. Now, that's a cartoon, but I always find it instructive to show this photo micrograph movie of what is happening. CAR T cell is in the bottom left. We're coloring tumor cells green. And what you're going to see over the course of 8 seconds is recognition of the tumor cell, activation of the CAR T cell, secretion of perforin and granzymes that will puncture holes in the cell membrane, and the tumor cell will explode. So here we go. And that tumor cell explodes. Now, this CAR T cell is a living drug. It will go and kill the next tumor cell. It will divide. Its daughter cells will kill more tumor cells. Now, speaking of anniversaries and 30th anniversary, the first proposal for a CAR T cell trial was submitted by a company called Cell Genesis to the U.S. NIH DNA Recombinant Advisory Committee in 1993. Here are the results of that first clinical trial published in 2000. We had a collaboration with this company on a way to grow the T cells, you can see. And this is in HIV, as Miguel mentioned, the connection. Adoptive transfer of CAR T cells in HIV results in long-term engraftment. But then, in the early 2000s, after a peak of inflated expectations, we reached a trough of disillusionment because in cancer, there were disappointing clinical trials, poor engraftment, some toxicity, and there was a wonder of whether this approach could really work. Now, fast forward to 2023, and we have in the US and EU 6 approved CAR T cell therapies in leukemia, lymphoma, and myeloma. We have TILs approved by hospital exemption in the Netherlands. We'll have an approved product next year in the US, also a T cell receptor therapy, allo T cells approved in Europe, and in some regions we have engineered stem cells for adrenoleukodystrophy and thalassemia, and we are scheduled to have the first approved CRISPR cell product and 2 products, hopefully in sickle cell disease before the end of the year. This is a slide from the FDA taking a wider view, not just of stem cells and T cells, but gene therapies in purple that are directly administered. These products are accelerating to be submitted for marketing application, and more are coming. So how do we track the expansion of this field? Well, one way is by looking at total CAR T cell sales globally. This is data from Bertrand Delsuc. And what you see, latest hard data for the third quarter of 2023 and estimated for the fourth quarter, almost $1 billion per quarter. And if one takes the estimate of the cost of the CAR T cell, since approval, more than 27,000 patients globally have been treated with CAR T cells. This does not include Chinese companies, and it does not include clinical trials. So I think it's safe to estimate that more than 30,000 patients have received CAR T cells worldwide. And we know that these CAR T cells can persist for years. Shown on the left is work from those early clinical trials with Cell Genesis, showing in HIV, CAR T cells can last for a decade. On the bottom right, from last year in Nature, is our trial in cancer showing decade-long persistence. And it's amazing to think, right? You deliver a drug, and 10 years later, by flow cytometry, you can detect that drug. The PCR is shown on the right, and enduring B-cell aplasia. These are CARs targeting CD19. Now, we know that not all patients respond, even in pediatric acute lymphoid leukemia with the best response rate. We know that we have challenges in solid tumors that I'll talk more about in a few minutes. How can we teach every T cell to be all that it can be? Well, solid tumors. unlike blood cancers, there are additional challenges. In blood cancers, we have pretty well-defined targets in CD19 and BCMA. In solid tumors, we have challenges of antigen escape, antigen heterogeneity, trafficking, and the extreme hostility of the tumor microenvironment. How can we think about improving the potency of T cells? Well, we can use different activators, cytokines, induce improved metabolism through transcriptional regulation, do gene knockouts, but which combinations? So I started tracking the literature, and these are a couple of papers from 2021, and then more appeared. And then the ASH meeting at the end of that year, I had a list of 24. And then Carl June asked me to co-author a book chapter, and we came up with a list of 40. Now, how do you know which of these combinations are best, right? Let's say just pick 2 combinations, there's 40, 2 to the 40th is 1 trillion. That's a very large number. So while the macroeconomic environment is challenging, the science is continuing to advance. So I think a challenge for the regulators is to think about the new combinations that are coming along and maybe multi combinations, and how are those dealt with in clinical trial permission to proceed. Well, if we think about designs and potency, this is from a review we wrote a few years ago showing 10 different models of CARs using different ways of targeting and enhancing. I want to focus on one, a so-called TRUC, that secretes anti-tumor cytokine. Now, the anti-tumor cytokine in this case is IL-18, and we're going to combine that with a CAR to enhance potency. This is data from Bilang Hu. You can see with the addition of IL-18, there's enhanced survival in a mouse model. There were reports from our group and others on using IL-18 to improve potency. Well, let's put IL-18 in a faster CAR. Why do we want to make CARs faster? Well, a few reasons. Here's data from our group looking at our conventional day 9 CARs and then shortening culture to 5 days, and they're more potent. And then if you shorten the culture to 3 days, they're even more potent. So we've combined the IL-18 approach with a 3-day culture in a clinical trial that's currently open at the University of Pennsylvania. And I'm showing you early results here, and I want to emphasize that the eligibility criteria, among the eligibility criteria in this trial, is that patients, had they been able to receive a commercial CAR T, as indicated by the label, they had to have received that commercial CAR T and failed. So every patient that you see in pink has received a commercial CAR T and failed: Kymriah, Yescarta, Tecartus, and Breyanzi. Every patient in green is a complete metabolic response. And I'll also point out we started at a very low dose here, 3 million cells, not per kilogram, 3 million total body cells. We've been doing dose escalation, but the results that we're seeing at these low doses are really, really remarkable. And there are other developers looking at shorter CARs. This is data from Novartis that's been previously presented at ASH and recently published in September in Cancer Discovery. On the right, the clinical response, the arrows are ongoing responses. On the left is the engraftment, and it may be hard to see on the screen, but there is the dose escalation there. They started at 2.5 million cells. The very faint gray lines are the commercial product Tisagen Leclusol, and you can see that the engraftment is comparable at a 25-fold lower dose. There's a Chinese company, Gracell, that is developing a 24-hour CAR. and showing some really impressive results. We have preclinical data ourselves on a non-activated 24-hour CAR. Now let's talk about QC and how we release the cells. What I'm showing you on the left is our conventional 9 or 10-day expanded CAR T, and we do the usual things: viability, phenotype, residual, endotoxin mycoplasma, vector DNA sequence, and so on. But for a short CAR, even let's think about a 24-hour CAR. How do you think about CAR expression where the gene may not be fully integrated? How do you think about vector copy number, replication competent lentivirus, and potency where it is certain that this product is still changing, and it will change when it is infused into the body? Now, this is a processing flow that could be applicable to many cell types. You source the cells, there's a culture you may select. Maybe there's gene modification, you're transducing or gene editing, expanding, and then harvesting, pulling, separating, and then formulating and filling. With the initial culture, let's talk about raw material. We know, because we derive our source material from humans, that not all collections and manufacturing runs are predictable. Perfect is the enemy of good enough, and the question is, what is good enough? How do you know that you always have the right cells and what cellular raw material is acceptable? So look at this color palette, and I'll ask you 3 questions. Are all of those colors blue? Are all of those colors blue enough? Do all of those colors have blue in them? Here's another way of looking at this. In the blue circles are successful manufacturing runs. In the red circles are failed manufacturing runs. We failed to make the dose. There's a correlation with CD3 percentage, lymphocyte percentage, and an inverse correlation with monocyte percentage. But where do you draw the line, and would you exclude patients below a certain line if in retrospect you knew that you could successfully manufacture a dose? What about those patients in the blue boxes? So how do you qualify starting material? Gene transduction and gene editing. We're currently using retroviral or lentiviral vectors, but to do gene editing, you want to do that without a viral vector. We have rapid evolution of gene editing tools and even epigenetic editing. So what are going to be the requirements for preclinical data, conduct of clinical trials, and follow-up studies using these tools? We have new technologies for viral-free cargo delivery, and we have to think about efficiency, lack of toxicity, yield, repeatability, and regulatory compliance of all of these technologies. Well, CARs are moving beyond oncology. We started in HIV, but there's some very exciting data being presented in autoimmunity. In fact, lupus, just this past week, 8 out of 8 responders. We have preclinical data on CAR T cells for fibrosis, even some potentially CAR T cells for aging. And T cells, of course, are more than CARs, T cell receptors and TILs. CARs are moving beyond T cells to NK cells, macrophages, stem cells, and derived from iPSC. And as I close, thinking about future prospects, will we remain in the autologous setting? Will we have off-the-shelf or allogeneic? What about both, and how is that possible? Well, what if we combine the CAR technology with the COVID vaccine mRNA technology? And that's what's shown here. So in this case, the mRNA in a lipid nanoparticle is encoding a CAR targeting fibroblast activation protein. And the lipid nanoparticle now is coated with an antibody that directs it to T cells in absence of the LNP homing to liver. This is a preclinical study in an induced murine model of cardiac fibrosis, showing the efficacy of this approach. And there are a number of companies, including one on which I'm a co-founder, that is developing this as an approach that could provide wider access, because this would truly be off the shelf. Now, I realize I'm close to time. I just want to very quickly mention how we're thinking about other ways of providing access by not only having these advanced therapies available at tertiary medical centers. Penn Medicine is a system of 7 hospitals, and we're thinking of a hub and spoke model where the academic center leads, and then we provide expertise to community hospitals that are able to accept, maybe not for clinical trials or maybe better for commercial products, but those community hospitals could potentially treat patients with CAR T cells or other advanced therapies, or perhaps after their infusions. And so in thinking about this infrastructure, we have our academic clinical trials, but we also have clinical trials from industry, and we have the commercial product. So the goal is to get all of our hospitals up to speed in being able to treat these patients and manage them throughout their infusion and beyond. So the key questions are standardization and operations, but how do you do that and give autonomy? Educating all of the clinical staff how to standardize that, collecting and analyzing the data, and assure that the new sites are performing at high quality, managing the cost, and doing that at a low cost footprint. So the challenge, as we see in early rollout, is we have so much coming along in research and development. We do have regulatory approvals, but sometimes R&D proceeds so quickly, or it's not keeping in mind the ultimate regulatory approval and commercialization, that there's a mismatch. So we need to ensure that we have communication up and down the line to ensure a smooth pathway. And what are some critical path issues for wider patient access? We need to work on enhancing potency, especially in solid tumors. Ex vivo manufacturing is complex. We need to implement automation, shorten culture, and perhaps in vivo. Education and training at all levels, including IACT and other societies. We have to recognize that there are disparities in geography, race, income, caregiver availability, and finally, these therapies are financially complex. Could we implement value-based payment that links price to income? And could we implement predictive biomarkers? So in closing, I'll acknowledge that I'm one representative of a very large team at Penn Medicine Center for Cellular Immunotherapies, led by Carl June, our collaborators at the Cancer Center at Children's Hospital, thanking our funders, philanthropy, industry, government, collaborators at the FDA, and especially our study participants. Many, many names. It truly does take a village. And with that, I'll stop and thank you for your attention.
Thank you, Bruce. Inspiring as usual. That's the purpose. I will hold off questions until the end of the panel, because Bruce, despite being up very early, will carry on being with us to join the panel. So thank you, Bruce. Hold on. And if there's some challenges, I'll WhatsApp you anyway. So this parallel channel of communication. So then let's go on to the panel, where we have 3 great speakers that will give us different perspectives. And at the end, I would like to engage with you in discussion. We're talking about bringing innovation into clinical value. The first speaker is Professor Luis Pereira de Almeida, who's professor at the University of Coimbra, Faculty of Pharmacy. heavily involved in different organizations in that university, in experimental biology, advanced therapies, and neurological disorders, and the innovative centers there. He's done extensive research on viral vectors, I believe we're going to be cell engineers, right? Cell and gene therapy is going to come into engineering cells for a purpose in vivo or ex vivo. And we will need different parts of that engineering. And the gene delivery which is carrying the message, and Luis will do that much better than I, is essential on that step. So, Luis, over to you. The way we will do this, while Luis comes, is there will be 3 presentations, and then at the end, the 4 of us will be there to entertain questions. But at this point, over to your presentation. Thank you, Luis.
Thank you, Miguel, for the nice introduction. It is a great pleasure to be here. Thank you to Fatima, to the president of Infarmed, Dr. Rui Ivo, and to all of you to listen. So I will try to, in these 10 minutes, do a little bit of history. And as you know, we are inheriting 150 years of science, of biomedical science. So since Mendel and Darwin, that we understand how the message was being passed, but it was really fundamental also the understanding of the DNA structure that materialized the information in the DNA. And then the possibility to manipulate this DNA with the recombinant DNA technology by Paul Berg, Herbert Boyer, and Stanley Cohen were fundamental also to get where we are today. I didn't have the time to put all the people that were really the pioneers in vector development. I will just highlight Jude Samulski for the adeno-associated viral vectors, but many others were extremely important, particularly for the lentiviral vectors, Inder Verma, Didier Trono, Luigi Naldini, and many others. And this got us to the first clinical trial led by French Anderson in 1990. And actually, the 90s were a very inspiring decade in the terms that a lot of clinical trials were ongoing. People were developing technology. The mapping of the human genome was ongoing. And people thought that each one of our genes could be used as a new drug. Now, the thing was, how can we deliver each one of these genes, these new drugs? And the answer came from nature, to use viruses. Viruses have evolved over millions of years to parasite our cells, and so they are the best machines to do that. And this is exactly what people thought, that we could use the viruses by taking out the genes involved in pathogenicity and splitting the vector genome in at least 2 plasmids, one encoding for the proteins that will assemble a viral particle, and another one with our gene of interest. Now, when we introduce this into cells, cells will do the job for us. They will transcribe and translate this packaging construct into proteins that will self-assemble to produce a viral particle that will incorporate the vector genomes. And the viral particles will be generated. They will be secreted into the media, or we can disrupt the cells to take it out, depending on the type of virus. Now, these viruses can be used to introduce our genetic material in the target cells in a very efficient and safe way. This can be done both for ex vivo as well as in vivo gene therapy. So ex vivo, we will take out cells, as we just heard before, and modify these cells and reimplant it into the patient, or we can introduce directly the genetic material to the human organism. And this can even be done for CAR T cells, as we just heard. Now, these vectors, we have an arsenal. We have different vectors that we can. I will not go through all the different kinds of vectors, but I will just call your attention to these 2 groups. We have integrating vectors when we want that the information gets perpetuated in the daughter cells, And we have non-integrating vectors such as the adeno-associated viral vectors that have emerged during the last years as extremely promising. And as well, on the integrating side, the lentiviral vectors have emerged as the most promising approach, I would say. Now, one of the things that makes gene therapy so appealing is that in many cases, one single shot is sufficient to have a permanent effect, or at least a long-lasting effect. This is an experiment I did myself in 1998, where I injected vectors in the brain of the animals, and you see we can detect the turn gene 1 week, 3 months, and 1 year after one single shot. And so this changes the paradigm of treatment. One single shot will be like a molecular surgery, ensuring permanent therapy for the patient. Now, unfortunately, the 90s, at the end of the 90s, we had 2 accidents, one that vitimated Jess Gelsinger, and another one that led to leukemia in 3 kids. And so this kind of put gene therapy on hold for some years. I would say that this first decade of the new century slowed down gene therapy, but of course this allowed the technology to evolve and to explore other applications and technologies, and it brought us to 2012 when the first gene therapy product was approved in Europe, Glybera, to treat this lipase deficiency. It was not a commercial success, but it opened the door for many other successes. Luxturna, we heard already about big success, preventing blindness in kids. The CAR-T, you heard already about all the different, the 6 approved products using CAR-T, and we'll have many more in the next years. This year, the therapy for hemophilia, something that people have been, trying to do for many, many years, and that finally was approved for both hemophilia type A and type B, also big success. All these were gene addition approaches, okay? So we can, besides the vector to carry the genetic material, we have to talk about the cargo. What are the vectors carrying? And what does this cargo do to our body? Well, most of the products that are approved are gene addition, okay? And these were examples that I showed of gene addition. But in some cases, what we need actually is gene silencing. And in fact, I've been working for many years in a disorder that is called the Machado-Joseph disease, or spinocerebellar ataxia type 3. And we basically have been trying to understand the basis of this disease and also to generate therapies. So this disease is caused by an overrepetition of this trinucleotide, CAG, that encodes for glutamine. This kind of mutation happens it is known in 9 different disorders. The most well-known is Huntington's disease, but here we have spinocerebellar ataxia type 3 or Machado-Joseph disease. And these overrepetitions of the trinucleotide will translate into repetition of glutamines within the ataxin 3 protein. So this mutant ataxin 3 protein acquires toxic properties and will cause neurodegeneration in different regions of the brain, where it will also aggregate. And these regions of the brain will be particularly those that are involved in motor control. And so the patients will develop ataxia, they will lose the control of the voluntary movements. And so this is a terrible disorder that is rare, but still prevalent in some regions worldwide and also in Portugal, where it reaches the highest prevalence in the Flores Island in Azores. Now, what we think we could do to try to block this disease, if we think at this gene that is being encoding for ataxin 3 that is being transcribed into a messenger RNA, translated into a protein that will aggregate and will cause the disease, we can try to activate the mechanism of RNA interference. And by doing that, we can get rid of this messenger RNA and prevent the production of the protein. And we've done this in a number of publications. Let me just show you an example where basically what we did was to inject vectors encoding for mutant ataxin 3 in order to induce the disease. And you see here the result, the aggregates of ataxin 3. Now, at the same time as we injected these vectors encoding for mutant ataxin 3, we injected vectors encoding for silencing sequences on the right hemisphere. And you see here the drastic reduction in the number of aggregates. So this seems promising. And also when we look at the motor behavior of the animals, you see that a non-treated animal can only equilibrate for about 20 seconds and the treated animal for about 60 seconds. So again, it goes in the same direction. It is an approach that we are further developing. This is an approach of gene silencing. Many people over the world are investigating about gene silencing. It is slightly more difficult to do it as compared to gene addition. But now there's this new kid on the block, the gene editing or gene repair. I don't have the time to go through all the process, but in general, if we can go a level upstream, so we can go directly to the DNA and try to do the gene editing of the DNA, and in this way we can prevent, for instance, the production of this messenger RNA. This is under study for many years, and we had the meganucleases, we had the zinc fingers, we had the TALEN, and more recently the CRISPR. And the CRISPR really took the place because they are much easier to generate on every lab that has molecular biology capabilities. I don't have the time to go through all these guys that were extremely important in making it a reality, the CRISPR. As you know, Emmanuel Charpentier and Jennifer Doudna received the Nobel Prize. Maybe it should have been shared also with other people. Feng Zhang was also very important. It was the first guy to demonstrate that this could be used for therapy in eukaryotic cells. So to make it very short, here with the CRISPR system, we only need two elements. Cas9 nuclease that will cut the DNA, and a guide of RNA. And this is what is different from the previous system. RNA, it is an RNA that will recognize your target DNA, and this is much simpler to generate. And so by doing this, we can induce a double-strand break in the DNA, and on the simplest situation, the cell will try to repair it by non-homologous end joining. And so this is a very error-prone mechanism of repair, and so typically it will add or it will take out a couple of nucleotides. And by doing this, it will induce a stop codon, and so it will basically inactivate this gene. And this is exactly what we did here, also for Machado-Joseph disease. We generated sequences targeting the mutant ataxin 3 gene, and you can see here on the non-treated side the aggregates of ataxin 3, and on the treated side the drastic reduction in the number of these aggregates. Again, this seems promising, but still don't forget, we have to address in gene therapy one of the major issues is delivery. Inder Verma said it many years ago. Gene therapy is all about delivery, delivery, and delivery. So getting to the right place in the right amount is particularly challenging. We are very interested in the brain, but this issue also appears in other organs. Now, in the case of the brain, we were fortunate enough that there was the discovery that a specific serotype of adeno-associated viral vectors is able to cross the blood-brain barrier. And so this was the work of Brian Kasper, Martin Barkatz, Mimoun Azouz, and many others. They showed, first of all, that the AV9 could cross the blood-brain barrier, and they later on they showed that this could be used to treat spinal muscular atrophy, making this a big success, the success that opened the way for treatment of the kids, and kids not normally on the most severe form would die before the age of 2, now are doing their more or less normal life. So this was a fantastic success that opened the way for many other treatments in the future. We have been also using this approach, and you can see here that the control animal will have trouble to complete this task, while the treated animal is able to complete it. And when we do the same test on the rotarod, it's again a very clear effect, so the control animal will fall while the treated animal will continue to equilibrate. So showing that, again, we can use a non-invasive approach to deliver the AAV vectors to the brain and to silence the expression of this mutant ataxin 3. Now, I showed you a very pink picture. Okay, everything is gonna happen, and but in fact, there are many challenges ahead. We have the challenge of the pre-existing immunity. Probably we all have antibodies in this room for AAVs. Some of us have sufficient levels to neutralize AAVs if we need a treatment with AAVs, and this is an issue that we are trying to tackle in this project, RDAT, basically by using different approaches such as the destruction of the antibodies by a specific treatment. Other issues are the small capacity of AAV vectors, very small vectors, 25 to 30 nanometers, and being able to carry only very small genes, 4 to 4.5 kb. So this can be an issue, but we can split this genome in different AAVs, and by doing this, we can overcome this. Problems of analytics, problems of price that we can discuss. I showed you some results from the people that are here. I don't have the time to go through all the people that are contributed in my group, in the collaborators, and all the people that contributed in the past. Yeah, just this was for next, that we are launching the 1st January. We will implement a new gene therapy center in Coimbra, where we'll try to tackle this by having a R&D unit, a production unit, and a clinical trials unit. And with this, I finish. Thank you very much.
To continue this panel, I will call to the stage the panelists, the moderator and the panelists, Isabel Vieira from Infarmed, Margarida Menezes Ferreira, independent consultant for ATMP.
Thank you, Luis, and thank you for, we're just making sure that we save time so everybody's on stage now. So we continue, and we'll come back to these different topics for the discussion at the end. And we now have Isabel Vieira that will talk to us from the regulatory perspective. Isabel is has got a PhD in development physiology and a degree in pharmaceutical sciences, and she's currently non-clinical assessor, and she's alternate member of the CAT. She will take presentation, and then we will stay on stage and follow it from there. So over to you, Isabel.
So, good morning. Oops. So, the European framework for marketing authorization of the ATMPs has been established in the ATMP regulation from 2007, which entered into force in 2008. So, in 2009, the Committee for Advanced Therapies has been created in order to provide expert, regulatory, and scientific advice to ATMPs. This committee integrates a multidisciplinary scientific and regulatory expertise covering quality, non-clinical, and clinical areas of assessment in order to be applied to the assessment of different types of ATMPs, gene therapy, cell therapies, tissue engineering, combined ATMP products. So, with respect to the pre-authorization tools that are in place to give some regulatory and scientific support to the ATMP's development, since the early phase of these developmental programs, I would like to quickly mention some of them, the most important, that are the contribution of the Committee of Advanced Therapy. The Innovation Task Force, the ATMP classification, the SME's ATMP certification, the scientific advice, and the PRIME support. So, for the task force in innovation, this is a forum for early interaction with developers. It's a new opportunity of dialogue on scientific, regulatory, and legal aspects concerning the innovative medicines. It's a multidisciplinary group, and the Committee of Advanced Therapies is involved in the briefing meetings with ATMP developers. The Committee of Advanced Therapies is also involved in the scientific recommendation on the ATMP's classification. So the companies can consult the CAT in order to confirm if the product that is under development can fulfill or not the scientific criteria to be defined as an ATMP, and in particular as a gene therapy, cell therapy, or tissue engineered product. So around 600 requests of scientific recommendation have been submitted since 2009, and mainly of the classification concern tissue engineered products, followed by gene therapy and cell therapy products. CAT is also involved in the certification procedure for ATMPs. So this procedure is focused on the assessment of non-clinical and clinical data that has been generated in early phase of the development of this ATMP. So, in 2010, EMA published in the website the first press release concerning the first certification procedure. This is a procedure that has been put in place in order to support the SMEs in the development of new ATMPs. Another tool is the scientific advice. So CAT is involved in all the procedure of scientific advice concerning ATMPs. So an increased number of scientific advice for ATMPs is noted since 2017. So in the last years, this number of requests is approximately stable, around 50-60 per year. But it's very important to note that an increased number of scientific advice concerning gene therapy medicinal products has been submitted since 2017. And this has a very important impact also in the marketing authorization that have been granted during this time period. Concerning the PRIME schema is also a very important tool for regulatory and scientific support of ATMPs. So PRIME is in place since 2016. It is expected to benefit more complex medicines and application with smaller data sets, which is the case of ATMPs and orphan medicinal products, covering a broad range of unmet medical needs. So an accelerated assessment is also available for products that are under this PRIME schema. And the PRIME provides a regulatory support and also a scientific advice for the products that are under the PRIME eligibility criteria. So if you look the primes that have been granted for ATMPs, we have around 45% of the procedure concern ATMPs. So last year, 2022, 8 prime designated medicines have been recommended for approval. 5 of them are ATMPs. Oops, sorry. Breyanzi, Carvykti, Ebvallo, Emgenix, and Roctavian. And in this full picture, we can see the importance of this PRIME procedure in order to support the marketing authorization of new ATMPs. So we have 12 products that have been in the market after the PRIME is in place, namely the 6 CAR-T cells that are in the market. All of them have been supported by PRIME. And we have also the press release for the EMA indicating that these 2 first CAR-T cells have been approved under the PRIME schema. This is Yescarta and Kymriah in 2018. So, concerning the marketing authorization, as you know, for this type of products, a centralized procedure is required for the marketing authorization of ATMPs. The Committee of Advanced Therapies plays a central role in order to assess the quality in non-clinical and clinical data based on a risk-based approach that has been implemented for this type of products. So the final opinion and the final authorization of these products is in the remit of the CHMP and the European Commission, respectively. So, in the annual report published by EMA for 2022, some medicines have been selected in order to represent the most significant progress in their therapeutic areas. And this is very important. 9 medicines have been proposed. 6 of them are ATMPs. And we have Emgenix and Roctavian. This is an in vivo gene therapy for the treatment of hemophilia B and hemophilia A, respectively. We also have Upstaza for the treatment of a rare, ultra-rare neurological disease. We have 2 CAR-T cells, 1 CAR-T cell anti-CD19 and 1 CAR-T cell anti-BCMA, and a somatic cell therapy. So this is this highlights the 6 products that have been in the market last year. So, concerning the gene therapy, we have 2 gene therapy approaches, and we have in the market products corresponding to in vivo gene therapy. And in that case, we have the introduction of the therapeutic gene directly in the patients, and we also have another strategy based on autologous use of this product based on the use of CD34 cells. And in that case, we have Zynteglo's, Streamvelis, Libmeldi, and Skyzona. Only 2 of them are still in the market, and we have the 6 CAR-T cells that are in the market. 4 CAR-T cells anti-CD19 and 2 CAR T cells anti-BCMA. And most of these products are first-in-class products and bring a lot of innovation to these new products that are available to a broad range of therapeutic indications. And I only include here some examples of the press release published by the EMA, all saying the first CAR T cell for different type of treatment, a new gene therapy, the first gene therapy, for example, for hemophilia A, the first gene therapy to treat a new neurological disorder. So these type of products bring a lot of innovation to the patients and to the market. With respect to the somatic cell therapies, the first 2 products approved are no longer in the market. We have Halofizel and Ebvallo that are still in the market. I put the Portuguese flag in Halofizel thanks to Margarida that coordinates the co-rapporteur team on the assessment of this procedure. And in last year, we had the first therapy to treat transplant patients with lymphoproliferative disease. For the tissue engineered products, we also have the first 2 products approved are not in the market, no, but we have 2 important products for autologous use that are approved. One of them has been classified as the first stem cell therapy that is available and recommended to be marketing authorized in the European Union. So this is the full picture of the products that we have in the market. During the first 5 years, only a few products come to the market, but in the last years, namely in 2022, we had 6 products that have been granted a market authorization. So the prime support play a very important in this profile of improvements. And I would like to mention that different types of marketing authorization have been granted for this type of products. For example, most of the products that have been recently authorized are under a conditional marketing authorization. And last year, we had 2 marketing authorization under exceptional circumstances. So based on this situation, the post-authorization regulatory tools plays a very important point because there are some efficacy and some safety studies that need to be conducted post-approval. The risk management plan need to be assessed in short timelines. And we also have, for some ATMPs that are in the market, new therapeutic indications that have been approved, namely for Yescarta and for Kymriah. So, in order to conclude, this is a long road. This is a continuous interaction with the regulators in order to follow the life cycle of the ATMPs. And I speak about the pre-authorization measures, and here I present the case of Kymriah. Kymriah was in the market since 2018, and Kymriah obtained a prime designation, 6 scientific advice in quality, non-clinical, and clinical aspects, and we have safety and efficacy studies that are still ongoing and are scheduled for December 2038, the assessment of safety, and, for example, studies that are ongoing until December 2023 in order to continuously assess the efficacy of these products. So this is a long way to work, and I would like to thank you for the attention.
Indeed, Isabel, I would like to thank you for a great presentation. And I just take a couple of messages there. One is cooperation and collaboration in the development. And probably a second one is a rule of thumb. Since 2017, 2018, 5 years, we had 5 products. In 1 year now, we have 5 products. So it is an acceleration. I mean, the numbers are not exactly that, but just the rule of thumb is accelerating significantly. And each one of those has a lot of learnings that in terms of staying, going away, and what happened to those products could give us a lot of learnings in terms of the experience that you have. But based also on that, really, if I can call upon you, I mean, indeed, we've been around the block for a while already, and I still remember a lot of the sessions we've done together a few years ago. Maria is a biologist, a PhD in medical biochemistry, worked in France, in the US, and has been with Infarmed for a long time, and now is helping anyone that needs help around here. And she'll tell us about their experience and their vision on that. Maria, thank you very much. I welcome you to the stage.
Okay, thank you. Thank you very much for the invitation. This is really great to be back home somehow. And I like to see many faces here that I know. So I have this difficult task of not reproducing the usual regulatory slides, because it is a different role that I have now. But I'm going to try at least to give some perspective what has been the issues with the cell therapies and gene therapies in the evolving. So when I started in this field in 2004, and at that time, as you may remember, there were lots of pressure on stem cells. Stem cells were the paradigm of all the treatments and everything that is going to be cured, and there were many incidents related to this type of approaches. And as you probably know, there's even a Netflix series now of The Bad Surgeon, which is Paolo Macchiarini that was doing synthetic trachea build-up with stem cells, and he killed several patients, and now he has a Netflix series, and he's in prison now in Sweden. So in 2023, he was finally sentenced to prison. So this was somehow the expectations and the difficulties we had at that time. Meanwhile, also in the many hospitals, there were experiences done directly with cells injected into the heart, so in what was considered the non-homologous use of cells. So, in fact, the Commission was really preoccupied and decided to launch a very important piece of the legislation, which was the regulation for advanced therapies, meaning that all these type of products would have to be authorized by the European Medicines Agency with all the experts, and created through this regulation the Committee for Advanced Therapies that was bound to do all the assessment work to be presented to the CHMP and then to the Commission for the approval of products. And also the Annex 1 that most of you know of the previous legislation for the pharmaceutical general legislation, where all the requirements were built, were revised to include a directive that had all the necessary information to be provided in a dossier for submission for marketing authorization. So those two pieces of legislation were very crucial for the launchment of the advanced therapies. And the regulation also recognized immediately the particularities of the advanced therapies domain, so meaning that you had to do you have really to be preoccupied with the use of not only live materials as medicinal products, but also materials that were not necessarily stable, that were even, when administered to the patient, had their own life on their own because they were dynamic materials. So this created a totally different perspective and created, for example, the need for specific GMP requirements, and also because of the long-living materials administered to the patient, also the pharmacovigilance and the long-term efficacy and safety follow-up were realities that had to be rebuilt compared to what it was even from the clinical trial phase to the marketing authorization. So all this was all built up also with many incentives for small and medium-sized enterprises, so that the try to push up the innovation also built in Europe. So this was really a very important document that is still, even with the new revision of the pharmaceutical legislation, which is now ongoing, is going to maintain. There's only a few changes that I'll mention in the end of my presentation. So I'm not going to go very much into, I was given 10 minutes, so. Prime is, in fact, Isabel already mentioned, and she mentioned extensively, has been crucial for the development of this type of products. Most of the products, this is an old slide, but it's still the same type of approach, have been crucial for the two main domains that Louise already mentioned, the adeno-associated viruses, which are non-integrative viruses that carry a cargo that are very important for the delivery of a gene, and the lentiviral and retroviral vectors, which are integrative viruses, and therefore they provide to a long-lasting generation of cells into the human body. So those are the two crucial areas that have been developed. Those are still ongoing, and there are many more efforts, like Bruce Levine said, also targeting, for example, solid tumors, which are a very difficult area to be tackled. So again, 47% of products have been supported by this continuous scientific advice system, so that they are made ready to go into the marketing authorization and to get into the market. And getting into the market does not mean that they reach the patient. That is going to be the afternoon, the next session, and I'm very eager to listen, because I think most of these products are not in Portugal. Very few. There's 2 of them, I think, from all of them. Oh, maybe Rui will update me. In fact, another approach that is very important here is the orphan designation, because most of these developments tend to go first, because they're first in gender generation of this type of products. They start with smaller populations, and therefore the orphan indications are very much sought. Most of the products that are on the market are orphan designated, and so I have in yellow the AAVs, and in green the lenti or retroviral vectors, and in blue, written in blue, or the cell-based, which are a minority also because of the complexity of the production system. So many are withdrawn, and some of them are withdrawn for marketing purposes. So they're withdrawn by the company because they don't find a niche to be provided. The prices are extremely expensive. And for example, if we go back to Hemgenix, it has been written in the press in the US that is the most expensive medicine ever produced. Of course, it is very important to take that into consideration for the budget of countries like Portugal, but sometimes you need to have restrictions and decision makers. Not only Portugal, but all over Europe, countries have that type of decision making. So as said before, the Prime, we have now 2 products that are ongoing in assessment, that at least is what it is given by the European agency. I only know that for certain that the exa-cel is now under assessment in Europe and in the US, and is a very interesting product, which is a CAR T cell that has been, it's not a CAR T, it's a CD34 plus cell that has been genetically modified by gene editing. So it's the first gene editing, again, first in class, that has been submitted to assessment. And it is very interesting. If you want to follow, you can see the discussion last week, I think, on the risk of off-target addition that has been discussed by FDA with their consulting committee, and it is a very intense discussion, really, and you can follow not only the innovation of the strategy itself, but the innovation of the analytical tools that you need to go in order to identify what is on target and what it is off target, and the difficulties of having the interpretation what off target means, because off target can be in silencing parts of the genome and not even and not having any impact. So it is something that is difficult to tackle, difficult to interpret, and certainly very difficult for a regulator to take decisions upon it. So the importance of experts for these type of areas is extremely important. So and now the gene editing is also progressing, and is progressing not only with the tools for analytical tools, but also to try to tackle the difficulty of the traditional gene editing, which is double-strand breaks of the DNA, meaning that you have repair mechanisms that can provide additional complication in the genomic expression, but also the need to have not interfering so much with the DNA, so to have just single-point alterations of the coding letters one by one, and you have this base editing and prime editing that are ongoing now as alternative processes. So this is an unstoppable moving into better products, and it has been very important. So back a little bit to the what is the context that we have. ATMPs had major developments. So from the very start, the Committee of Advanced Therapies, and now I'm going to make my own publicity of the committee, were instrumental to do a special guide for GMP for ATMPs, which tackles lots of the particularities of the products, and it has also built the case for the revision for orphan designation, so that it could accommodate easily without having exclusivity granted, for example, for one lentiviral vector versus to the other lentiviral vector. So we needed to have some more opening for this type of protection. So there was also a revision of the definitions of the orphan designation and the orphan regulatory framework. So this is everything we did from these early days of the 10 years I was at CAT. And we were very preoccupied to create the conditions for the existing. So, for example, in relation to the cardiac repair, we created the condition for non-substantially manipulated cells, because that is the interface with transplantation. So we needed to have an integration, what it is non-homologous use, and we created the risk-based approach. It is very important to highlight that the entire legislation for the advanced therapies is based on risk-based approach. So it was the first time in the pharma legislation there was the introduction of the risk-based approach, meaning that you can adapt the information you provide with the conditions of the risk, and you can justify your approach by a risk assessment system. So we did also advise the guideline for genetically modified cells, also to integrate the genome editing approaches. We are now revising the safety and efficacy of follow-up. Just to tell you, for example, for the gene editing, the follow-up now is at least 15 years. So that's a long way to maintain information going from a marketing authorization starting point to the holding of responsibility. And we are now also finalizing, or they are, because I'm not no longer there, the advanced therapy investigation of medicinal products to create a tool to enable the easy access to regulatory framework of those companies that are not necessarily big pharma. Most of the companies dealing with this type of products are not big pharma, are small companies that, of course, their aim is probably the phase 2 to be sold to a bigger one and have the product really launched at a planetary level if possible. So just a little word on what is going on now, because there's a major reform of the EU pharmaceutical legislation. We already mentioned it, and it is a very important piece of information because it's trying to streamline and to accommodate many of the requirements from industry because of the complexity of the European system. I don't know if it's going to get there, but let's hope that we'll get a little bit better. So there's a new regulation that also aims to integrate all the system that was in place for the centralized applications and the directive that will build in and whatever was already in the Annex 1, and it's going to take, for example, in advanced therapies, the directive that has all the Annex 1 information for the marketing authorization is going to be built in, but it has a provision so that the Commission can create a system to revise even this requirement. So this is an ongoing for later stage. Because I'm making my own statements, I decided to be open in my own statements. And in fact, I have concerns on this pharma regulation that is now eliminating the existence of CAT with all the work that CAT did and the particularities of the domain. I don't know how far the CHMP will be able to tackle with all the other medicines, because being 45%, there's always the 65% that take a lot of time also at the CHMP. And is it going to be able to really deal with all the instrumental materials that were developed by a group of experts really dedicated only to ATMPs, and how far this is going to be maintained with everything that has been done and needs to be done all the time. And importantly, the disappearance of the Biologicals Working Party, where I belonged for 20 years, and that has done by CHMP mandate the assessment of all the quality of the biological products. And now the BWP is going to be reduced to an expert group, meaning that only a few member states are going to be nominated and some of them will remain out, which means that the connection between the quality, the inspection, and the OMCL networks become a little bit more impaired. And this is, for me, a high preoccupation, and that's why I expressed that already to the Commission when there was time for comments, and I did those comments, but they're lost in space. So I needed to say that. In the revision of the pharma regulation, there's a very important aspect that has been solved or tried to solve, which is the assessment of the genetically modified organisms, which is a very important issue for Europe. It does not exist in many other areas of the world, but in Europe is very important. And it was a tremendous difficulty for the development of products in Europe during the investigational phases, because you have to go into all the authorities for the environment and have specific permissions by each of them with different requirements, etc. So now, even at the level of the investigational product, the submission for the GMO authority will have a centralized assessment, like you have it for the marketing authorization. So it reduces the burden. That is a very important issue that has been defined. One concern, again, of mine is that the classification of the medicinal product and ATMPs is going to rely still at the EMA, but it's an EMA obligation, is an agency obligation, is not necessarily a CHMP obligation. And I don't know how this is going to be handled, but I hope it will be handled correctly. The good thing is that it's still the European agency that has the mandate to classify as medicinal product and advanced therapies and consult the tissues and cells authorities that are in the transplantation domain. So this is quite a good aspect. In what concerns the pharma directive, it's a preoccupation that hospital exemption is maintained exactly as it was. Hospital exemption means that an advanced therapy medicinal product can be approved nationally for a specific patient use with less requirements than the marketing authorization, meaning that it competes in the market with lower prices to the marketed products. And this has been one of the problems, for example, with one initial cell product that was based on chondrocytes that did not have market because most of the market was occupied by a hospital exempted materials. So this is a preoccupation that is going to be again built in. There's many discussions on the data protection, and industry, of course, is very preoccupied that the data protection is going to be reduced to 2 years. So it will going to be 6 + 2 years instead of 8 + 2. But an interesting issue, it has this Article 26 that has additional master files, meaning that you could apply for the certification of a master file of substances that are biological and substances that are provided in the market, in the manufacture of the product, not necessarily the product itself. So this means that, especially for advanced therapies, is important because sometimes you use feeder cells or you use certain type of materials that could be certified instead of filed all the time for the same product. So I think this is a most welcome. I've done a thorough understanding of what is the legislation. So I think this is really interesting. And now finally, the new substances of human use, which is the legislation applicable to transplantation. It has been also revised and is now is going to be launched. I think it will be applicable from next year. But has one very difficult situation for the development of investigational products in Portugal and in Europe, which is said by the import and export of the starting materials, because cells are starting materials that have to be collected in Europe from patients that will have to be sent somewhere and exported to be manipulated, and then the product is important. The importation of the product as medicine is covered by the medicines domain, but the export of the cells to be manipulated should be the sole responsibility of the manufacturer and not of an entity ruled by the authority of transplantation. And this is a preoccupation because some parts of the development phases, and especially early development phases, remain confidential. This is my last slide. Remain confidential. It is important that this confidentiality is maintained because it has selection criteria of donors, etc., that has to be taken in consideration. So this is, for me, another preoccupation. And because I'm me, I decided to put this here so that I can have people supporting me.
Great presentation. You touch on a series of topics that will come back, particularly on the regulatory point of view. I'll make a few comments and a question there. But I would like to, before opening to the audience, get a bit of a warm-up, couple of questions, then come back to some of the regulatory elements, and then onto the public. We are going to be around for another 10 minutes or so due to the delay of the start anyway. So to engage Bruce, that is probably sunrise has already happened there. I would, to the panel and to Bruce, how do we see the future about, it's a bit of a trivial question, but behind it comes a lot of elements, the autologous versus the allogeneic and the opportunity of manage this through gene editing. So I'll start with Bruce, and then we want Luis to make a comment about that, and if our two regulatory person also make a comment. This is really just to get the discussion going before we go into the hot stuff you mentioned, Margarita. Bruce?
Yeah, thank you, Miguel. I hope you can hear me okay.
Yep.
So I'll start with a few comments. You know, there is an argument for allogeneic to have something off the shelf, but there have been challenges, and the clinical data has not, I would say, been all that has been hoped for effector T-cell therapies, CAR-T, CAR-NK. It certainly has a role because there are patients from whom we cannot generate autologous cells or get enough cells. But I don't look at it as an either or. I think both may persist in the long term. But to add some concerns about allogeneic, there are a couple of adverse events in a trial in Australia using transposons with a T-cell lymphoma that originated from the product. There was a paper this week that came out that showed that extended culture of CAR T cells may reactivate HHV6, which may be associated with some neurotoxicity. That's not to say that adverse events cannot happen with autologous therapies, but autologous therapies, and you saw that I mentioned the 27,000 number, there have not been genotoxic events associated with autologous retroviral or lentiviral modified cells. CRISPR base editing to be determined. And to wrap up to your question on the potential of in vivo, you know, a few years ago, I would have been much more of a skeptic, but I was sitting in this chair reading Science magazine on the report of the Intellia trial in transthyretin amyloidosis. And then you see the data and it's dose response, and you think, well, maybe this really could work. And we see some more evidence since then. So I think long-term, prevalent diseases, global access, we're looking at some version of in vivo.
Thank you, Bruce. Luis, do you want to?
I tend to agree with Bruce. So from a researcher point of view, of course, doing in vivo gene therapy, and so skipping this need to take out cells, modifying it, evaluating the cells and putting it back, which is a logistics nightmare, it would be much more interesting. And it was science fiction until recently, but there is some evidence that it might be possible. And the thing is, the tools that we have these days are becoming much better. So, for instance, with the AAVs, we can engineer the AAVs to specifically transduce only one cell type or one organ. And this will make it possible to do direct modification of T lymphocytes or other cell types without the need to take out the cells, to isolate the cells, and to do all these procedures. But in the meanwhile, we have a technology that works. The CAR-T, it's an extraordinary technology, so we should take advantage of it, and in parallel try to develop the other approaches, such as the allogeneic as well as the in vivo approach, I would say.
Before I hand over, Luis, can I ask you a question, and I'm going to put you on the spot. If you had a magic wand and could say, okay, I'm going to build the ideal gene delivery tool, A, B, L, and P, what would it be in 2 words? If you could just say, right, this is
so the
iPhone of cell therapy.
It's very difficult to answer. You know that there has been this kind of split between the non-viral people and the viral people. The truth is the viral vectors are still more effective. The non-viral approaches nowadays are becoming better, and also they have the opportunity with the gene editing that they don't need to be as efficient as they needed before. Because if you have a system that basically modifies the genome permanently, you don't need to be that efficient in terms of constantly delivering a nucleic acid cargo. And so I think there is an opportunity, especially for the gene editing tools, for the non-viral systems. Still, the viral vectors, I think, are the system that at present is the most efficient one. And in the middle, we have also the viral-like particles that I think seem very promising. We have the extracellular vesicles that also are systems that are in between the synthetic and the biologic viral world, and that can also be very promising, I think. They have shown very good results. So we have it's not only black and white, non-viral, viral, we also have, besides the systems in between, like the extracellular vesicles and the viral-like particles, I would say.
Yeah. I'll let Margarida then Isabel make a comment on this, if you have.
Oh, I have comments. I have always comments.
I know that. We've seen it.
I think in the end, everything that is biological tends to be replaced by chemical. That's the trend of manufacturing easiness, of manufacturing consistency. So in the end, and we see it with the vaccines for COVID, you had the adenoviral approach and you had the mRNA approach, and certainly the mRNA approach took the lead because it was easier to handle. Vaccines is a particular case because you don't need a huge amount of material. It's sufficient that you trigger the immune system and then the immune system does the job. While for a product that has to interact with the human body, you have a problem of dosing. So as non-black and white, I think if the dosing can be made correctly without any toxicity, which is a problem with the particles approach and the chemical approach of liposomes, for example, if it can succeed, you may have a difficulty with the dose, the targeting the dose and the delivery of the cargo. And the viral vector has this fantastic advantage that is a live material, so somehow it disseminates and it can be more or less directed. So I think both will survive, both approaches will survive. But the tendency will be to become chemical.
This 20 years ago, 10 years ago, is a scientific fiction discussion, but yes. Do you want to make any comment?
Yes, we are facing new products based on mRNAs, based on this CRISPR-Cas, and it's a learning process. We have to deal with this type of assessment and to see new products coming to the market. And for us is a challenge to assess this type of new products.
Sure.
So the regulator need to follow the science always.
You made a fantastic review of all the experience, and you highlighted one point along your presentation, which was the collaboration and the discussion between the developers and the regulators, because we can only do this together. We're getting on to the time, and I wanted to make a comment and bring in Bruce from a US/FDA experience, as well as here, and you're a bit outside Louis' perspective on this. You made a series of comments, Margarida, on the legislation and the evolution of the legislation. I'll make my own comments on top of that, because and that will probably prompt the discussion. First one is about CAT, and we have several people here involved in CAT. When I saw that the first time, I said, well, I'm a bit unhappy with this. At the same time, this is a sign of maturity. Do we need to keep them outside of the mainstream, or should it come to the mainstream? But if it does come to the mainstream, then the mainstream needs to be able to deal with it appropriately. So that's my take, but I would like to have views on this. The SOHO, I'm actually quite comfortable with it in general, I think, but maybe your experience can tell differently. The GMO, for me, is an idiosyncrasy, right? It should have been solved by now, right? As I said several times before. And it was solved for a period, so why did we came back? The hospital exemption, it's going to be here to stay. I think there's a lot of elements that are making it better, and different players are impacting on how it should be. I think we have an opportunity to get the right balance, and I hope it will be. And then finally, and that's where Bruce comes in, master file, standardization, and what the FDA now is coming on the platform approach, which is kind of something that will be modular for different products. Easy for the same developer, more challenging across developers. That's where master files could, or elements like that. There's discussion on AAV as a platform, iPSC as another platform. So comments on this, I'll probably let both of you to start, then maybe you can talk a little bit about AAV if you want. And then I think Bruce very closely in contact himself and through ISCT with Peter Marks and the FDA may have a comment on that as well. So maybe starting with you as well.
It's very hard to address this type of strategies because there are strategies that are taken at different levels. And my level as an assessor at the national agency and also collaborating as an alternate member at the CAT, from my point of view, our experience is gained with the work that we have been developed during all these years in CAT. I'm a recent collaborator, but Margarida was in CAT since the start of the committee. So as a sort of beginner, I would like that to explain that it's a gain of experience and we can contribute with this experience in different scenarios. So this depends on the structure that we need to contribute. And at that moment, the work at CAT is a very productive work. All the people are aligned in this type of products. We have experts from different fields, and usually we collaborate in this type of discussion with these different point of views. It's like a puzzle in order to build the whole picture of the procedure, and it's very important. I don't know how it can work in other contexts, because I know this context is difficult to extrapolate, but I believe that EMA have enough experience to give all the support and to maintain this way to work, because it's important to work using this strategy.
Thank you.
And we can see the results last year is very productive work in terms of ATMPs coming to the market.
I mean, the work is there, the value is there.
Yes, it's my point of view.
To harness and make sure that it is well framed on going forward. Really, a couple of words, particularly if you want to stress the master file.
I think the important information is that in Europe is not allowed to have a master file on a biological substance. That's the bottom line, what it is. We never accepted, and it's not possible legally. So in this revision of the pharma directive, it is possible that the EMA provides a certification for a biological substance that can be the active substance, as well as a supporting manufacturing product, meaning that you may have biological reagents, for example, that are used in the manufacture that can be certified, and therefore being certified, you no longer have to present in every file every information on the safety on that reagent. This alleviates enormously the burden of a file. So this is the aspect, the legal aspect. On what it concerns platform manufacturing, this is a reality that has been already ongoing for monoclonal antibodies, and we're used to it as an approach. The problem is the filing itself. So I think platform manufacturing of AAVs is a reality that is there already, because manufacture of active substance of AAVs are 3 or 4 that produces for all of them. So in a way, you have platforms all over. So it's easy to be handled as a platform, and you can build information. The problem is the flow of information that has to be legally solved.
Thank you.
Yeah,
Two words?
just 2 words about this. The platform, for me, makes all sense to have a couple of AAVs which we agree that they will have an expedited approval, because in fact they are all the same, only the transgene is being placed. For me, it makes all sense.
Thank you. 2 words, Bruce, and you will close it.
Well, it's hard to do in 2 words. Yes, FDA is working on the harmonization. I will perhaps say a word about the accelerated pathways. I think there is some discussion now on gene therapy drugs that are going through the accelerated pathway, where the data may or may not be there for clinical efficacy, which gets to this right to try impetus in the US. And I think as developers, as regulators, we all have to be very careful to provide access, but access to safe and effective medicines. Because if things go over the line, then that impacts the validity of the development of these therapies and the integrity of the regulatory oversight process.
Thanks, Bruce. So now that the panelists made a few comments, I'll open up to questions before going from the audience. And we have a microphone there, 2 microphones there. We have an excess of microphones. No, we have a question. Can you identify yourself?
Hello, good morning. I'm Carla. I'm working in the SME in Portugal. Actually, we are manufacturing in vitro medical devices, and special for companion diagnostics. But I have more a general question, not in particular to the advanced therapies, but how can we participate in Portugal as a volunteer in clinical trials? Is there any option in Portugal to participate?
I'll probably have Isabel comment, but I think there is all the opportunity to participate, and there's quite a bit of experience. I was impressed to see, for instance, the use of alofisel as one of the products, and other products there as well. I think I'll just take advantage of one comment. There's challenges about clinical trials, and then you can actually prepare your answers. There's challenges about clinical trials across Europe in making sure that we have the right number. I don't think Portugal is in any way less available or less suitable or less disposable to actually have those clinical trials.
No.
I think to do a clinical trial, there must be a sponsor, and the sponsor needs to be convinced to do that in Portugal. And I think we all have the responsibility to make that availability and that proposal. And that if the sponsor wants, it will happen.
The purpose of a clinical trial is to demonstrate something on safety or efficacy of the product. So it has a use of that information. The inclusion criteria is the most important item that the sponsor uses. So the sponsor is the one that is going to decide whether you, as a patient, is valuable and you are in the inclusion, follow the inclusion criteria to be used as information. So the major decision is not on a voluntary basis on your side, is really the sponsor that can accept you, patient, as being...
Yeah, and you should lobby the sponsor.
So you have to be aware, and normally patients are aware of what is going on, or can be aware of what is going on.
I'm talking about non-patients.
Just volunteers, but those are specially designed that also relate to the inclusion criteria of the patients because of the healthy volunteers. But that's a decision, again, related to the quality of information of the disease itself, even if you are not the disease part. the disease arm of the trial.
Thank you. Maybe there's another question? Yes, one back there.
Thank you so much. This is a question for Professor Bruce. My name is Vitor. I'm a student at the Faculty of Pharmacy of the University of Lisbon. I don't know if you have watched the movie Ford versus Ferrari, but my question is very simple. Given your perspective in terms of a person who's very much on the lab and on the investigation side of things, what would be one change that you would propose that would kind of be a win-win situation for the regulatory approval of drugs?
Well, that's like picking your favorite child, right? Maybe I'll mention something that we haven't talked about yet, which is the evolution of analytics. At least in the US and in the Code of Federal Regulations and elsewhere, we have reference to analytic methods that are many decades old. I'm talking about thioglycolate and other methods for sterility testing. We have to get to the point where we have more rapid analytics to decrease the turnaround time of these products. And another challenge of analytics is developing the analytics for these new gene editing methods and assessing on target, off target, and also when we have that data, understanding what is and what is not important. That is the virtually impossible from the potential to have an adverse effect. So I'll keep the answer short in this case, but there, Peter, are many other answers to your question. And I think that is the value of having so many developers. When you look at the number of clinical trials, just of CAR T cells, it's close to 1,000 around the world. And we add in all cell and gene therapies. I showed that slide of the number of parameters of improvement. So I think it's important to continue to have parallel development and to share information. Thank you.
Thank you, Bruce. And then I think the next big development is going to be precisely on managing all that big data into improving how we go forward. But I will propose to stop there. Thank very much the panels, the audience for engaging. We continue. We have a break that, as we start a little bit late, expanded a little bit, we're going to delay the break, and we would like to see you back here by 11:40. So that we go on to the session, and Fatima will take over from Mia Ben. So thank you very much. And
We are back and we are going to continue this event with session 2, Health Technology Assessment, Patient Access. We will start with the panel Deploying Clinical Value to Patient Benefit, moderated by Fatima Ventura from Infarmed. I call the moderator and the panelists to the stage. I call Martina Schussler-Lenz from the Paul Ehrlich Institute, Cláudia Furtado from Infarmed, Francisco dos Santos from Crioestaminal, Filipa Moita from the Portuguese Institute of Oncology in Lisbon, and Mencia de Lemos Belmont from the Spinal Muscular Atrophy Foundation.
So, I'm higher now. So, I welcome you back from the break. I hope you enjoyed the networking outside as much as we enjoyed to have heard the first session. Bruce Levine giving us the background and the future of CAR T cells. Luis Almeida showing us the promises of gene therapy, and also we experienced to hear the experience from Isabel and Margarida on the regulatory aspects of ATMPs. But research and development and regulatory approval of these products only makes sense for the general medicinal products, of course, if the final product reaches the patients. And patient access has several phases, starting from the developers who invest in clinical development of these products, to the clinicians who use these ATMPs in their patients, and who gave them the difficult and to have the difficult decision on the eligibility of a patient to these treatments. Also to the health technology assessors and reimbursement authorities that have to judge the place of the new products in the context of other alternatives, if they exist, in the market. Finally, because the patients are and should always be at the center of all the decisions taken at the level of regulatory and of the stakeholders that work with the regulatory authorities, it is essential to listen to patients, to engage them in all the life cycle of the medicinal product, from the development to the decision making. So in order to help us at the discussion on all different aspects, but interconnected aspects of these ATMPs, we will have in this session 5 excellent panelists that will bring us their view on each of the aspects we selected for this session. And I would like to introduce the first one, which is Dr. Martina Schussler-Lenz. She will be the key speaker for this session. Martina has graduated from medical school at Mainz University Hospital in Germany. She worked as a clinician scientist in Berlin, New York, and Barcelona, and completed her fellowship in internal medicine, hematology, medical oncology at the Mainz University Hospital. She joined the pharmaceutical industry in 1992 and was responsible for several clinical drug development projects of anti-cancer agents. Since 2005, she's working as a medical doctor at Paul Ehrlich Institute in the Department of Hematology, Cell and Gene Therapy, where she reviews advanced therapies in clinical trials and marketing authorization, as well as provides advice to ATMP development and life cycle management. She is committed to facilitate the development of ATMPs and has a specific focus on providing guidance to academic developers of ATMPs. Dr. Martina has served up to February this year as the elected chair of the Committee for Advanced Therapies, CAT, at the European Medicines Agencies, and she has done that for 2 mandates since she was appointed at the Paul Ehrlich Institute in February 2017. So I invite Martina to come here and tell us about the...
Wow, thank you very much for this very kind introduction, and thank you so much for having me here. I think it's a great honor for me to celebrate with you 30 years of Infarmed and also to celebrate ATMPs. So that you have made that to one of your topics, I think is really very great and is very remarkable. It's a little bit of a challenge to talk as a clinician and a regulator about a lot of things that have already been mentioned in the previous session, regulatory frame. So I'm trying to put a little bit of a different aspect on it, and will also include the aspect of patient access. So from research to market-approved ATMPs and patient access, what we hear, what we know, and what we expect. What we hear, ATMPs and specifically gene therapies deliver with a one-time administration a causative treatment with long-term efficacy and potential for cure to patients in need. We hear also that ATMPs are the most expensive treatments, and you see here Hemgenix, $3.5 million, the new treatment for hemophilia B. And the question is whether the cost savings for the overall healthcare system by generating factor IX levels over years will compensate for that. We know about Skysona, a $3 million ATMP for the treatment of cerebral adrenoleukodystrophy. And the interesting thing about Skysona and also Synteglo is that for these two cell-based gene therapies, they were first authorized, we authorized them first in Europe. And then they were withdrawn from the market for reasons that you can imagine. And later on, the company Bluebird filed the marketing authorization, and they are now licensed in the US. And then there is this ATMP, this gene therapy, Zolgensma, with a price of 2.26 million. That's a price that was in Germany in mid-2020 for the treatment of spinal muscular atrophy. As other ATMPs administered by one single administration, and in this case, a single intravenous administration. And well, for that spinal muscular atrophy indication, it was the first gene therapy and it got a lot of attention. Another treatment was already available, Spinraza, where you see here from the German data that I could retrieve, anticipated around 500,000 treatment costs per year, but a treatment that needs, in small children, repetitive intrathecal administration. And then the new treatment, Erisdi, which is an oral drug, and you see here also the estimated treatment costs. Just to put that a little bit in perspective. What we also hear that innovative payment models are needed to address the value of ATMPs for these products that have a single one-time administration and then an expected duration of efficacy over years or even lifelong, and that patients in the EU have different levels of access to innovative treatments. Now, what we know, and that's the main part of my presentation, and I will show you give you some information about marketing authorization, clinical evidence at marketing authorization, scientific considerations, and patient access to ATMPs. So, starting with the ATMP regulation, I'm not going to repeat that, you've already heard it from the previous speakers, that we have these three groups, that they are expected to go through a centralized marketing authorization and then available in theory on the EU market in all the member states. The exception from that centralized marketing authorization was also already mentioned, but for completeness, I want to mention it here as it is a national legislation, an exception that empowers the European member states to authorize manufacturing and administration to patients under certain conditions, and the conditions are mentioned here. And there is a lot of talk about it. You've also heard about that is going probably to be kept with the new pharma legislation with some amendments, and it's what is probably something that you would judge as positive for member state-based production is probably criticized by the companies because they may be worried that there is so much CAR T cell, in that case, production locally, nationally, that it puts them into problems. But we'll see how that it will turn up at the end. And just to mention that one CAR T cell that has been a lot in the media also, that example from Spain. When you look at the data, you will see that the clinical development for that CAR T cell, there were a very limited number of patients that were actually treated under hospital exemption, and that the majority of patients were treated in clinical trials. But yes, there are now more attempts and more plans to get the CAR T cells in the hospital exemption, and we will see what happens with that, and it will probably also be a part of the discussion. But now getting back to the centralized marketing authorization, and you hear the medicine and the ATMP development cycle and the life cycle, starting from discovery to preclinical discovery, mostly in the hands of academia, very important. Then the clinical evaluation, clinical trial authorization with the CITES now, that puts maybe developers, but I know it for sure us at the agency, Paul-Ehrlich Institute, we struggle a lot with the CITES system, and it has not yet, in our view, there are a lot of improvements needed to make it a smoother type of procedure. We have all the different types of national and EMA support in terms of scientific advices. And then at the level of the marketing authorization, already also alluded to, the CUT, so other committees like PRAC and COMP involved in finally the decision on benefit risk assessment, the opinion, which is then passed over to the European Commission for the actual marketing authorization. And then there are different types of approvals: full approval, conditional, exceptional circumstance, are also already mentioned. And then the very important and a little bit different for ATMPs compared to the other non-ATMPs is the importance of the post-authorization time, the post-approval time, it's not only pharmacovigilance, but it's really the phase where the product is authorized, but where for the ATMPs, we need more data. We need more data from the patients that have been enrolled in clinical trials. So that's the clinical trial follow-up, because at time of marketing authorization, we usually have a limited follow-up. And you or HTA may say, why did you do that? You have only a 2-year follow-up for a CAR T cell, for patients in a CAR T cell trial, and only a 2-year or 3-year follow-up for the patients enrolled, the hemophilia patients treated with the AAV. Why don't you have, and you cannot say, you don't have the information at that point whether CAR T-cell treated patient will be continuously in remission. We've heard about living drug and long durations of response, but still there are uncertainties. And for the AAVs, the big question, is there a dilution of the effect? And I will come to that later. What about, is there a cure of a hemophilia patient or not? And why are you basing your benefit-risk assessment on data that do not give us that information? But that's a big question, but it's also, in a way, not so difficult to answer, because the question is, can we come to a positive benefit-risk assessment in the specific indication, considering the totality of the data that we have, or shall we wait for a 5, 6, 7-year follow-up, which means that this gene therapy is not available on the market? It's always a little bit that balance that makes the ATMPs very specific, and that makes the post-authorization phase very important. Also, we always have obligations, we give obligations to the marketing authorization holder with the marketing authorization, not only that he needs to qualify the treatment centers for CAR T-cell or CD or human stem cell modified products, not only qualification of the treatment center, and I also will go into that a little bit, but also to do a safety and efficacy follow-up, and ideally to include each and every patient that is treated with an ATMP in a PAS or a PAES study based on a registry. Very difficult in the European situation. I have no time to go into that, but it's very difficult in terms of where do we have suitable registries for example, beta thalassemia, in which countries are these registries linked. That's really something that need to be solved so that we can generate post-authorization better information, a clinical follow-up on the patients treated with ATMPs. Now, the evidence at marketing authorization, as for other products, we need robust evidence at marketing authorization. And when we define clinical evidence as the totality of clinical data about the use, benefits, and risks of a medicinal product across the development program, we should be aware that these includes clinical trial data. That's the mainstay for us for the benefit-risk assessment. But it can, of course, include data from hospital exemption, which we have seen, but which are usually real-world data with all their disadvantages. But also data from clinical data, early access programs, clinical data to inform on the natural cause of disease. All of that is clinical evidence. And and we are aware that the randomized controlled trials are the standard for providing robust and confirmatory evidence, and there are a lot of guidelines, and there are also guidance from Europe and from the FDA that say that substantial evidence should actually be provided in 2 randomized controlled trials, but there are certain circumstances, and that's in these guidelines, where you can build on 1 instead of 2 randomized controlled trials. We'll see later whether that comes true for the ATMPs, but in principle, the same principles apply for ATMPs, randomized control design, also in late stages of non-Hodgkin lymphoma, for example, randomized to best supportive care or investigator's choice, and it is composed at the cut on quality, non-clinical and clinical aspects of medicinal products containing genetically modified cells, and it has a specific annex for CAR T cells. You have seen these data already in a different format. So these are the approved ATMPs by somatic cell therapies, gene therapies, tissue-engineered product with the first control select in 2009. And we see this shift now from the tissue-engineered products and somatic cell therapies towards the gene therapies. And you see that since 2018, except for one all the products that have been authorized in Europe, all ATMPs have been gene therapies, and not all are on the market. Now, the principles of gene transfer, I wanted to briefly mention that because I think the understanding of the different principles of gene transfer in vivo, the AAVs, recombinant adeno-associated viral vectors, where the genetic correction comes with the AAV on one hand, and then the ex vivo genetically modified or gene-edited cells. That understanding, I think, is important also when we talk about value and when we talk price and when we talk about these things. So knowledge transfer, I think, is important. And I have given here the example of the CD34 hematopoietic stem cells, and that also, like the AAVs, are meant to do a genetic correction of a severe, of a genetic disease, and very different from the CAR T cells that are adaptive immunotherapies. I'm not going into depth into the CAR T cells, but rather we'll try to give you some examples on clinical data for the other two groups. You see here the authorized CAR T cells, and they are now have gone very nicely from authorization in last line treatment, third line non-Hodgkin's lymphoma, to earlier lines of treatment based on randomized controlled trials. So we have seen the first, and we have assessed the first CAR T cell randomized controlled trials. Very, very interesting, and so there are more CAR T cells now coming towards at least now second line treatment. And then the CD34 hematopoietic stem cells, where you see here Zynteglo's Caizona and Libmeldi, and it just coming back to the mechanism of action and to what it involves. And you see here in the figure, starting from the bone marrow harvest or the leukapheresis to the CD34 cell purification, genetic modification or gene editing, cryopreservation, quality control, and reinfusion to the patient, and then importantly, safety and efficacy follow-up and the clonal tracking of vector insertions. And then the question is, what does it require? It requires manufacturing, of course, and quality control and all of that. But it requires, in a country that wants to use these treatments, the correct diagnosis, diagnose a genetic disorder, neonatal screening, which has become very important for spinal muscular atrophy, for example, where it has turned out that Zolgensma is now available or approved and available in 45 countries worldwide. And genetic screening has made a big difference because you treat the children very early. Then to identify the correct patient population, and in this case that I will show you is Trimvelis, these are patients that have no HLA-matched stem cell donor and therefore have no adequate treatment option, and are then a patient that could be treated with such a gene therapy. The identification of a qualified treatment center, and that is then doing HSC mobilization, apheresis, myeloablative conditioning. So for these type of treatments, it will always be in principle a transplant center. You need infrastructure for acute and long-term monitoring, data collection and reporting, ideally good over Europe. You need logistics for ATMP handling, freeze, thaw at the treatment site, and logistics, of course, a lot of logistics that come from the companies that ship the samples to the US for manufacturing and back as it has long been done for the CAR T cells. Now we fortunately have more production sites for our approved CAR T cells in Europe also. And then, of course, you need all the authorizations. And this example, scientific and regulatory considerations related to efficacy and potential for cure, on this product that was already improved in 2016 by a major contribution from the Naldini group and Alessandro Aiuti, who was a part of the CAT also, and who had treated patients with ADAS-KIT, a fatal disease, a very ultra-rare fatal disease, 50 children per year in the US and Europe that have less than a 2-year survival without treatment, and that only part of them have a suitable HLA-matched stem cell donor, and the treatment that was approved then, Strimvelis, in 2016 was for patients that have no HLA-matched stem cell donor. And the therapeutic principle is that there is bone marrow engraftment of the genetically modified cells. The patient before undergoes a myeloablative chemotherapy. Then there is bone marrow engraftment and repopulation by the genetically modified stem cells, polyclonal differentiation, and hematopoietic and immune reconstitution in these children that, through the ADA deficiency, have lymphopenia, have a non-functioning immune system, and are dying from severe infections. And the data that were submitted at marketing authorization came from an open-label single-arm trial, 18 children, mean age 1.5 years at, not transplantation, at treatment. And then there was immune reconstitution in all subjects with a median follow-up of 7 years at the time of marketing authorization. So that was already quite a long follow-up. And we imposed with the marketing authorization then the condition to follow up all patients in that trial and also all newly treated patients. And we have this continuous yearly or sometimes 5-yearly reporting from the marketing authorization holder to the CAT, to the EMA, and the rapporteurs assess it and the PRAC assess it. And what we know currently is that altogether 35 patients have received this treatment, and there is 100% survival with now a follow-up of the longest patient of 20 years. So that's one example. And the other example that I wanted to give you are the AAVs, and there the example of hemophilia A, this X-linked factor VIII deficiency, where, and that's a little bit not so common when we see a new gene therapy, but was a special case here, there are quite good treatment options for hemophilia A and also for hemophilia B, the replacement treatments and others. And that is always a special, challenge then also in the benefit-risk assessment for this therapeutic principle, which is very different from the the genetically modified stem cells. With the AAV as a non-pathogenic replication defective virus, the transgene of interest is carried to the target tissue, and that the duration of efficacy, in this case production of factor VIII in the liver, that's a target tissue, is related to the maintenance of the AAV or the loss of the transgene. It's usually, the AAV is episomal, it's not integrated, and there can be loss, and we know that, and we've seen that, and you see it on the right side of this clinical trial where there were 134 patients included with a mean 2-year follow-up, and it was a non-randomized trial, but with an intra-individual base. So you have that patient with hemophilia, you follow him, and then you follow him for 6 months or 12 months, and then that patient enters the clinical trial. So you have the factor VIII levels before treatment and the bleeding episodes, annual bleeding rate, and the same efficacy readouts after treatment. And then you see whether through the AAV treatment, the gene therapy treatment, there is a reduction in bleeding episodes and the maintenance of the factor VIII levels. But here you see after some time, and this is up to 2 years, that there is a certain reduction of these factor VIII levels, and we know the reasons for that. And there is a lot of science about working on improving AAVs and improving their maintenance in the human cells. And so in that case, we did involve an ad hoc expert group with hematologists, hepatologists, and hemophilia patient representatives. That was on the level of the cut that we said, yeah, we want to have an AHEC group and we want to see what they say in terms of benefits. That is a very formal consultation, but a consultation that we now have with each and every marketing authorization, that patients are asked what is important for you when you have the disease. And the conclusion was that their supportive conclusion that they thought that the benefits outweigh the risk and that there is a relevance of these treatments, even if we don't know what happens beyond 2 or 3 years. So these are the marketing authorizations, again, summarized. Just to point out, they are, since 2018, the gene therapies, all orphan diseases, and you see types of approval. There are a lot of conditional marketing authorizations, and actually 7 out of 14 of gene therapies have been conditional marketing authorizations, which corresponds to accelerated of the FDA, and which implies that the data presented are non-comprehensive and more data need to be generated post-authorization, but we have already a positive benefit risk assessment. So what follows from there? The benefit risk assessment follows specific rules and guidelines, takes into account the specificities of the ATMPs, requires robust clinical evidence, but it's a case-by-case decision. It may be based on non-randomized single-arm trials, and there is a new guideline for that. It involves patient and physician organizations to get their view, and it requires always post-authorization data to expand the understanding of ATMP safety and efficacy, which is only possible if the ATMP is reimbursed and used. So that is a catch-22. If the ATMP is not reimbursed and used in that country, in Europe, in a lot of member states, it means that we don't have this incremental knowledge about the ATMP that we need from the patients treated under real-world conditions. But it's, of course, the first step towards patient access. And this is a little bit showing what happens in Europe in terms of market availability. And the source is a summary from FPR Weight Indicator 2022, where you see that of all orphans, and I show that because, as I've said, the ATMPs recently are all orphans, you see that an average of 39% of those that have received a marketing authorization are on the reimbursement list. We're quite lucky. Germany is, they can enter the market very, very fast, but now we have also, the German HTA has also imposes conditions, but they can enter the market and they start being reimbursed, and then in Germany there is a re-evaluation by the HTA after 1 year. You see that the time to availability in Europe ranges, after approval, ranges from 55 days to more than 1,000 days. And in some countries, these are never available, and you see here the CAR T cells. The highly variable availability, this is from a presentation at the CAR T cell meeting in Rotterdam, where you see non-availability here in red for many CAR T cells in the Eastern European countries. So, from market-approved ATMPs to patient access, of course, we have to acknowledge the different tasks and responsibilities. Benefit risk assessment, as already mentioned, follows certain rules. And then the added value and cost effectiveness assessment of ATMPs, very different, because benefit risks, we leave, of course, cost effectiveness completely out. that by the national HTA pricing and reimbursement bodies relies on comparative data, and there is a lot of criticism that ATMPs are not approved based on randomized controlled trials. They follow, the HTA assessment follows specific models that may not be suitable to appraise the value, clinical, economic, social, and mental of ATMPs, and also requires post-authorization data to support cost effectiveness and alternative payment models. So you see that this post-authorization data from the regulatory perspective and from the HTA perspective is needed, and there is a way where through early dialogue we could improve that, also make it easier for the companies, the marketing authorization holder, by having a very early dialogue on our requirements, on HTA requirements, and try to come to a common agreement, at least on the post-authorization phase. So, my last slide, what we expect, a favorable European environment for research and innovation that supports ATMP development, a favorable environment for patients to benefit from equal access to safe, modern, and affordable medicines, and that's the wording from the EU pharmaceutical strategy, improving the pre-planning of post-authorization evidence generation needed by regulators, HTA bodies, and pricing authorities to reduce evidence gaps and foster timely patient access, and reduce the administrative burden for companies and strengthen HTA capacity building and joint clinical assessment. And that's the wording of the HTA regulation. And with that, I close. Thank you.
I will open the questions for Martina as a keynote speaker at the end of the panel. From what we just heard, it's a very good bridge to the next speaker, which is Claudia Furtado. And in order to understand, to explain to us better why, as a regulator involved in the marketing authorization applications, and why sometimes the work we have done before doesn't reach the patient in the end. So Claudia has a degree in pharmaceutical sciences from the University of Lisbon and holds a PhD in public health from Nova University from Lisbon. She is the head of our health technology assessment, price and reimbursement division at Infarmed. And that is the division here that is responsible for HTA evaluation, pricing and reimbursement of not only medicines, but also medical devices and health products. She's also the head of the Information and Strategic Planning Division that is responsible for monitoring health consumption and expenditure and for defining and evaluating policy measures. She is currently assistant professor at the Portuguese National School of Public Health. And Claudia, you will have to.
Thank you, Fatima. And I hope that with my 10-minute presentation about the HTA and prayer perspective, I will not decrease too much the enthusiasm that we are experiencing this morning with the HTMPs. So, but the reality is that we have to face that access to HTMPs includes several dimensions. And there is no real value of granting a market authorization if we cannot secure the HTA pricing and reimbursement for that medicine, and also if that medicine is not available in the specialized centers to be prescribed. So I think it's very important that we focus on these dimensions, because it's only with the integrated approach that we can have access to these medicines. I don't know if all of you are familiar with the HTA process, but for those who are not, the HTA process is an instrument that supports pricing and reimbursement decisions and helps to maximize health gains for the whole population in a context of finite resources. So we have to choose, and we have to choose wisely when, where we allocate the resources that we have and for which we all contribute. So the pharmacotherapeutic, the HTA process includes a pharmacotherapeutic assessment that starts with the definition of the PICO, that is a population intervention comparators and outcomes. And based on this PICO, we are going to assess the evidence that is submitted by the company. And we assess the quality of the evidence submitted, and we assess the relative efficacy and safety. And we, after that assessment, we appraise that evidence and define if it has added value, if it has equivalence, or in some cases, if added value was not proven. So this is a pharmacotherapeutic assessment, then precludes the pharmacoeconomic assessment, that where we make an economic study where we quantify the benefits of these drugs and the costs of these drugs in comparison with the treatment that we identified with comparator, or in the absence of that, best supportive care. We also analyze the budget impact of this medicine in the health system. So based on these two dimensions, we have to balance how much gain are we getting with this new drug, and in comparison with what we already have in the system, and at the same time, how much cost it will represent in relation to that additional gain. So when we are dealing with HTMPs in this HTA process, we are faced with several challenges presented here in the previous presentations about the regulatory pathway. So consider, if even in the regulatory area there is some uncertainty, when we are approaching the HTA, when we have to make, at least intend to make comparative analysis, this is quite difficult. So most of the drugs are presenting, not only in Portugal, but in all Europe, several challenges. And from the experience that we have, and just commenting on the comment that Margarida made about how many HMPs we have in Portugal, we have already reimbursed 6 HMPs. We have 2 under assessment, but there are some that did not submit the process in Portugal, nor in most of the European countries, because even the companies sometimes recognize that the evidence that they have is not enough to make HTA and pricing and reimbursement decision. So we clearly have here, in some cases, a disintegrated pathway, because we have granted the market authorization, it is available, but no one can have access. So, and why is that? Because we are facing a lot of clinical uncertainty, lack of appropriate comparator, that's obvious, surrogate indicators, lack of long-term efficacy that Martina also presented, but also the colleagues from the previous panel, and of course the reduced number of patients that was somehow expected. This high clinical uncertainty is added by a high economic uncertainty, because we are dealing with medicines that, I don't know if it's all development costs, but I think now they are fighting for which one is the most expensive drug in the world. So we have a high price of that medicine, which reflects, when the economic study is done, in high relative cost effectiveness ratios that are not affordable for our health systems. Adding to that, most of these drugs are single administration, so it is supposed to have an upfront payment of 1 million, 2 million, 4 million, and I think it's all getting increased. This upfront payment associated with the single administration, and besides that, we have administration costs, and in this case, they should not be neglected, because the hospitals have to have the infrastructures, they have to be trained, and that should not be a part of this decision. However, and as we all see today, there is potential benefit to patients from these drugs. in some cases, and so, some probably not, because and they withdrawn the process in some of the HMPs that were assessed. But in some cases, we recognize that potential value, and you want, as a health system, to promote the access to those medicines, because especially when we are dealing with high unmet clinical need, we want to promote access, but we have to take into consideration this high clinical uncertainty and this high economic uncertainty and the context of a finite budget. So these reimbursement decisions are getting more and more difficult for Portugal. That is a country with not so much resources, but it's difficult for Sweden, Denmark, Germany, and the higher GDP countries in Europe. So how are we going to overcome these challenges? We are going to keep some of them because it's not possible to overcome all the challenges with pricing and reimbursement process, but we try to overcome these challenges by using some measures that are at our disposition. However, I would like to make it clear that despite having some measures to overcome these challenges, companies must strive to collect and present the best evidence possible. We can be more adaptive to the situation, but companies cannot be dismissed from the onus of presenting the best evidence possible, just to be clear. But let's see how we can overcome these challenges. And because I only have 10 minutes, probably now 5, I will just present them in a quick way, and then we can discuss it. Anticipation, joint assessment, conditional financing, monitor long-term outcomes, and better preparedness. In anticipation, I think it is the best way to address the limited evidence to support HTA. Companies should engage in early dialogues with regulatory and HTA and payers' body. Isabel mentioned the scientific advice. Martina also presented that this as a solution that is essential, and companies do not have any excuse to not use the instruments that are at their availability in both regulatory and with HTA regulation with joint scientific consultation. They should anticipate the needs of both HTA and regulatory agencies and develop clinical studies and evidence generation plans in accordance. Now, how we as HTA and payers can do our part. I think joint assessment will be an important part. It will not solve all the problems, but it will be an important part in addressing methodological challenges on assessment, because we will make synergies in efforts and expertise between member states. We all know that these medicines require some knowledge of how they work, and so if we gather the expertise that exists in these 27 member states, we will probably make better decisions. Avoids duplication of efforts by companies, because they only submit one dossier. We guarantee the involvement of patients in the process, because in some countries there is that involvement, in some countries there is not that involvement. So with the HTA regulation, we have that secured. We'll hope that it will be provided a timely assessment, and as we all know, the priority will be into 2025 for HTMPs and oncology drugs. So this might be a way of addressing the methodological challenges on assessment. On relation to price, there is a way, that is the conditional financing, to address upfront high payments and uncertainty on effectiveness. There is for instance, what we have used is phased payments dependent on results, with a lower initial payment, because I would like also to stress this. The conditional financing does not mean that we are going to pay that 2 million or 4 million euros per drug. We should negotiate the price of the drugs and make it a fair price. But even if it's a fair price, it should be phased depending on the achievement of the outcomes that is that are supposed to be obtained. So because with these HTMPs, we expect long-term benefits, sometimes life-term benefits, and we are paying at one specific moment. So we want to delay that payment in phases that guarantees that we are paying for what we were promised, as we all do in our lives. And we think that this lower initial payment and phase payment will help to reduce the uncertainty of upfront payments on single administration drugs. However, it will require registers, and in some cases it will be more burden to health professionals and also for us at agencies. In Portugal, we, for these drugs, CAR T cells, we have the payment conditional to overall survival using an already existing register that is the National Cancer Registry. For Zolgensma and also for the other drugs, we developed a specific register in Portugal for those drugs, and the payment is conditional to predefined outcomes for a determined period of time. This is also one struggle or that we have when we conditional the payment to outcomes. We have to agree on the outcomes, and sometimes that's not very easy. Or we have a case that was not possible even to continue. These registers will also help us to monitor and reassess, and these drugs are clearly the ones that need to have this monitor and reassessment of the value. To address the uncertainty on effectiveness and cost effectiveness, we should collect real-world data and generate real-world evidence to support the continuous monitoring of the effectiveness and long-term effectiveness, and, if needed, reassess the reimbursement decisions. Because we made the reimbursement decisions expecting some outcomes, if they are not achieved, or even if they are achieved, we should reassess those medicines. And for instance, for the spinal muscular atrophy, we have more than 150 patients included, and this helps us not only to assess the value, but gain information about the story of the disease, the standard of care, and the definition of comparators for newer drugs that are coming into the market. And I will finish with the more or less the same suggestion to overcome this challenge, that is anticipation. But in this case, anticipate to be better prepared, to address the challenges that we are facing in a timely way. We are in Infarmed very supportive of through the Horizon Scanning Initiative to identify emerging health technologies that may have a significant impact on health systems. And we should focus on those that address high unmet clinical need, because these are the ones that are going to have an increased demand by patients and by health professionals, and also those that have high costs. And because the access does not end in the reimbursement decision, we should use this information to prepare the introduction of these technologies with the development of registers if we need to collect additional data, thinking about different pay models, and I think it's quite important in this case to prepare hospital centers in training in infrastructure, because sometimes the drug is already reimbursed and is not able to be prescribed by medical doctors in Portugal because the structures are not certified. And with this, I end, and I hope that we are not very demotivated with the need of having cost-effective drugs in our systems. Thank you.
Thank you, Claudia. Now we will have the perspective of the developer in all this picture of the ATMPs and the way of making early access of such important products. So I give the word to Francisco Santos. Francisco is currently the Director of Production and Development at Criostaminal, where he is responsible for the cellular therapy laboratory activity. Francisco holds a PhD in biotechnology from Instituto Superior Técnico, and he was the co-founder of Cell2B Advanced Therapeutics. His professional activity has been mainly focused on the development and manufacturing of ATMPs for the last 12 years, and it's an example of Portugal is contributing to ATMPs development.
Thank you very much. Good morning, everyone. I'll start by acknowledging Infarmed for organizing this very interesting event, and also, of course, for inviting us to participate. We might be a strange body here in this panel, but what I'll try to show in the next few minutes is our modest contribution to improve patient access to advanced therapies here in Portugal. Most of you probably have heard of Criostaminal before. We have been operating for 20 years now. Our core activity is actually stem cell banking, so we offer the services to our clients to store their stem cells from multiple sources. We are the #1 banking company here in Portugal, and also we are part of the #1 group in Europe. But our goal has always been to be an active contributor to the development and patient access to advanced therapies here in Portugal. And a few years back, we come to realize that our banking activity was not sufficient for us. So we're being approached by clinicians, we're being approached by companies that wanted us to help them in some way to take their technologies to patients. And we actually are also quite looked for by patients that are looking for alternatives for their conditions, but those we always direct to the clinicians. And in fact, we come to realize that in this gap, there was no support system for this, both clinicians and small companies that wanted to take their products forward to do clinical development. And by that, I mean, most of them did not even realize the regulatory framework where they were in, what took to develop an ATMP and manufacture that in conditions that could be accepted for clinical development. So we come to initiate 5 years ago a secondary activity based on the development of advanced cell therapy products, where we basically operate as a CDMO for researchers, clinicians, and small companies, very much based on a strong R&D partnership network. And eventually we come to realize that now we have a small ATMP portfolio, although that was not our initial goal, but I'll talk about that in a few minutes. Just for context, our core activity is based on our cryopreservation laboratory. This is where we do all the activity of processing and banking cells and tissues, in particular umbilical cord blood and cord tissue, and then also adipose tissue. And what we come to realize is that even though we have been releasing these type of samples, in particular umbilical cord blood for transplantation settings, we are in fact a large bank of starting material for new innovative therapies that are currently being developed or will be developed in the future. So part of our activity of banking is actually to process and store these cells and tissues in the best way that they can be used in the future for future therapies. We also, of course, have a quality control lab that was initially designed to support our banking activity, but now is also able to support the development of ATMPs in our company, where we have an array of standard analytical methods for this type of products, ranging from microbiology, serology, flow cytometry, of course, microbiology detection, human viral pathogens detection as well, and a strong cell culture lab. And finally, our last operational unit is the cell therapies lab, and this was created 5 years ago. This is where we base our CDMO activity for our clients and our partners. We pretty much operate as any other CDMO, focus on offering service for product and process development. And this is quite important, especially for the type of partnerships and clients that we have, these clinicians that want to take their cells to bedside, or even small companies and academic groups that basically we support them to move from a lab scale to a manufacturing scale. We, of course, provide the quality control services by developing and validating analytical methods. We implemented and internalized things like sterility, endotoxins, and mycoplasma testing according to the GMP and following European pharmacopoeia compliance. This was something that was not easily available here in Portugal, for instance. And of course, we also manufacture the IMPs, particularly ATMPs, in our GMP-certified room. And for this meeting, it's also important to mention that as a company, we always have invested a lot of our R&D focus on cell therapies. And I bring you just a few numbers, not a complete of our portfolio, but for instance, just to give you an idea, since in these last 15 years, we had at least 8 major projects focused entirely on cell therapies, and these included things like new processing methods, new quality control methods, but even developing ATMPs for clinical trials and hospital exemptions. From that, we actually registered 4 patents on processing and catheterization of umbilical cord blood cells for clinical applications. Of course, it is based in a large partner network, and it includes both academia, hospitals, and a few SMEs here in Portugal and in Europe. And this is probably the most interesting number for this audience, is that from these projects, we are able now to develop, or are developing, 4 different ATMPs for different clinical conditions. And I'll give you just a brief overview of what we are doing so that you can have an idea that we are working with different type of products from different sources. And this came all out of our R&D projects and partnerships. So just to give you an idea, we have developed and manufactured also an autologous bone marrow CD34 positive cells for the treatment of acute stroke. We have also developed an autologous cord blood total nucleated cells for newborns with hypoxic ischemic encephalopathy. And both these projects are quite interesting because we developed these products. It was not our initial idea, but these were clinicians that approached us and said, I saw that this type of therapies is being done in the US or in Europe. I'd like to have the similar opportunity to offer that to my patients. Can you provide us with this type of ATMP? And also we have, for instance, an allogeneic expanded core tissue mesenchymal stem cell product, mostly focused on inflammatory and immunological diseases. All these 3 ATMPs have ATMP classification. And most recently, it is still in very early stage of development. We are developing now an autologous expanded chondrocytes product. And the first 3 products that I've just shown have already entered or are entering some type of clinical development stages. And this is just to give you an idea of our very small portfolio that we have, but mostly focused on the partnerships. So our allogeneic core tissue MSC product has already been approved for an hospital exemption in IPO in Lisbon to treat acute graft-versus-host disease. This was in 2020. We are also working on submitting a clinical trial authorization with this product for ARDS. This is acute respiratory distress syndrome. And this is a lot based off what came out of the COVID pandemic stage, where these cells were shown to have a clinical potential, a therapeutic potential for acute patients. And we also have been working on preclinical studies for other diseases such as lupus, scleroderma, and multiple sclerosis. Part of this work is actually already published for scleroderma, where we have these in vitro studies with patient samples where we can evaluate the impact of our ATMP in the activation of the immunological system. The autologous bone marrow CD34 positive cells for stroke has been approved within a phase 2A trial here in Portugal early this year. The sponsor is Centro Hospitalário Universitário de Coimbra, and we hope to be able to manufacture soon for this trial. And finally, the autologous cord blood total nucleated cells product for the treatment of hypoxic ischemic encephalopathy was also approved within an hospital exemption last month, if I'm not mistaken, also at the Coimbra Hospital. So we hopefully will all start to manufacture for these patients. Just to sum up and as a take-home message from Crestaminal, what I want you to realize is that, in fact, we are positioning ourselves not only as a stem cell bank, but as a partner for ATMP development. So we have been working together with the academic and medical community in the development of new and innovative ATMPs, particularly for unmet clinical needs. We are supporting SMEs in the clinical translation of their technologies. All of this we do with a very strict focus on trying to manufacture ATMPs at sustainable cost for healthcare systems. And finally, and ultimately, of course, what we want is to improve patient access to the advanced therapies in Portugal. And this is it, and I'll be glad to answer any questions afterwards.
So we just heard the first beginning of ATMPs as approaching, trying to facilitate patient access in some way. So this the beginning of all the beginnings. But now I would like to invite Filipa Moita. She will be giving us the perspective of the user of an approved medicinal product. ATMPs in this case. So Filipa Moita is a medical doctor, graduated from a medical school from the University of Lisbon in 2006. She was trained as an hematologist at the hematology department of Portuguese Oncology Institute of Lisbon, where she works since 2008. She's also an invited professor at Nova Medical School since 2015. And she has worked as a transplant specialist at the hematopoietic transplant unit in IPO Lisbon. And she currently works as a consultant at the same institution for this field. She's an active member of the Portuguese Society for Hematology, the Portuguese Lymphoma Group, and the European Mantle Cell Network. She's interested in the biology and treatment of lymphomas and participates actively in clinical research projects in this field at national and international level.
Thank you so much. And I'd like to thank you. 50% of the patients. And what we know is that in about a third of the patients, the responses are still ongoing. And the disease-specific overall survival is around 50% as well. And what is interesting in these curves is that after the first year, there is a plateau in the curves, which means that some of these patients are potentially cured by this treatment. And when we compare to the scenario that we had prior to CAR T cells, this is what we had. So for the same population that was later included in the ZUMA-1 trial, we had over a median overall survival of just 6 months. So if the patient failed 2 lines of treatment in the LBCL, there was no way we could treat him, and CAR T cells completely changed that paradigm. So this has been reproduced with other CAR T cell constructs, and again, for complete remission patients, we see the same scenario. And this has also been confirmed by real-world data. And this is a pooled analysis of several real-world registries, and as you can see, with several patients included. And what we can see is that both the efficacy is very comparable to the clinical trial pictures, but what's even more interesting is that the toxicity is even lower in the real-world scenario, as this is because we are gaining practicing in managing this toxicity. And what is also important is to know that in the real-world registries, there is a significant number of patients that would never be included in the clinical trials. And this shows us that we can do CAR T cell therapy for patients that are very different from what we could do with stem cell transplant, for example. We can include elderly patients, as this other analysis showed. We can do it in patients that have comorbidities, and so it's very difficult to establish a limit for which patients can benefit from these strategies. And as it was mentioned before, when we have an effective therapy in a multi-treated patient, we try to bring this therapy forward in the treatment, and this is a randomized trial that compared CAR T cells with standard therapy, which in this case was high-dose chemotherapy and autologous stem cell transplant, for patients that were primarily refractory or that had early relapses. And what you can see is that CAR T cells not only improved the progression-free survival, but also the overall survival. And this has become standard of care now for our patients. And this has also been reproduced in another randomized trial. And even now, for first-line treatment, this was a phase 2 trial of a very difficult to treat population, of a very high-risk lymphoma that are very unresponsive to conventional treatment. And in these populations, CAR T cells led to a high rate of complete remissions and also long remissions. And this is now being tested in a randomized trial, and this could again challenge our first-line treatment for these patients. What I've shown you for diffuse large B-cell lymphoma, it's reproducible in other lymphoma types, and this is mantle cell lymphoma. Again, with CAR T cells, we have median overall survival that is 3 times higher than we had prior to this therapy. And the same for follicular lymphoma. Again, there is a plateau on the curve, which means that maybe some of these patients are cured by this treatment in a very pre-treated population. And again, the same for multiple myeloma. So I'd like to show you now our experience in our center. So we started using CAR T cells in the end of 2019 with Axicell, which was the first approved. Then in 2021, we used for the first time Brexucell for mantle cell lymphoma. In 2022, we used Tisacell. And this year, we use Axicell for follicular lymphoma in an early access program. So in Portugal, we don't have the approval for follicular lymphoma yet, and we also don't have the approval of CAR T cells for multiple myeloma. And this is our experience. As you can see, well, the start of our program was a bit compromised because it coincided with the beginning of the pandemics, and we had to stop the program because there was a high uncertainty of how we could manage these patients. But after that, we restarted the program, and from today, we have treated 40 patients with CAR T cells, and 20 of these patients, they were treated just this year. So you can see we are expanding the number of patients that we have treated. When we look at the toxicity, we have results that are quite similar to what is published. And also, as for response, we see that most of our patients attain a complete remission and that the PFS is in keeping what has been published as well. Even though this is a very short follow-up because most of these patients were treated in the past few years. So we've mentioned this before, that now CAR T cells will probably be used for other targets in hematological malignancies, but also for other targets in solid tumors. And this will expand even more the number of patients that will be candidates for this treatment. And what we can now think is how can we deliver CAR T cell therapy to all the possible candidates. We have to think of the financial burden, as we spoke in this previous session, but also the complexity of the procedure. I'd just like to show you what the patient goes through once he is referred for CAR T cells. And he has to go through all of these stages, and in every stage there are some challenges involved. So we are treating patients not only from our center, but also from all centers from south of Coimbra, and also patients that come from Madeira or Açores. So this implies that the patient comes to our center to be treated and stays in our center for a long time. The bridging is a very important part because between the collection of T cells and the infusion of T cells, we have at least 4 weeks. And for an aggressive disease, this means that we have to keep the disease control in these 4 weeks. It doesn't seem much, but for a diffuse large B-cell lymphoma, it is enough for the patient to die in this time frame if we don't treat him with something else. And then we have one of the most important parts, which is the post-infusion surveillance. And the need for hospital admission, the need to keep ICU reserved for these patients. We have to be aware of the risk of acute complications, the risk of infections, and also in the long-term follow-up. These patients, they have a very prolonged B-cell aplasia, and they are the risk of infection, it can go on for many months and even years after the CAR T-cell is done. And also, I would like to say that there is also a very important role of the caregiver that sometimes needs to put also his life on hold, because these patients need to be constantly monitored even when they are discharged from the hospital. So this is a very high burden for the patient and his family, and we need to think of all of that. And I think that for the future, because we will have so many patients that are candidates for this treatment, we have to consider patient selection and also novel strategies, and we've spoken about some of these, like having locally produced cards or allogenic cards when the research has some better results. And I'll finish here. Thank you so much.
So, and as we come forward to the end of this panel, I was listening to Philippe, and I was thinking that we started in the first session with a kind of scientific fiction ideas, and we are becoming more and more to the reality. And for our last speaker, who is the ultimate reality, is the patient and their perspective. And more and more, we have to listen to what they think, their own knowledge about the diseases and their perspectives in so many issues. So I invited Mencia de Lemos. She is a member of the CAT, of Committee for Advanced Therapies at EMEA, where she represents patients. She focuses on rare diseases, and as a member of the CAT, she assesses clinical trials and real-world evidence to ensure that data capture is meaningful to patients, and this is really important. Mencia is also interested on registers and databases, capturing data in a way that it's fit for regulatory decisions. Prior to her engagement with European medicines agencies, Mencia was the president of the SMA Europe, an umbrella organization representing 25 national patient organizations across Europe that focus on the spinal muscular atrophy. In fact, Mencia lived the transition of treatments in this disease, in these neuromuscular diseases, from having no treatment at all to having already 3 disease-modifying therapies approved, being one a gene therapy. Thank you.
Thank you. So first of all, I would like to thank for inviting me and for taking into account the patients and making the effort to have the patients in the middle of the discussions. It would be excellent if all actors would take us in account, as the EMA does and as Infarmed does. I'm going to be speaking about access for patients to gene therapy, taking the example of SMA, because it's the one that I know more, because I have 2 specimens at home living with SMA. Both my children are affected, so I know the disease very well. So I'm going to start just to very briefly pointing out some aspects of the disease. SMA is a rare disease, it's neuromuscular and it's progressive. It causes muscle weakness and a very high dependency and a discapacity, but it preserves all the intellectual functions. So typically, people with SMA are people that are very bright, that want to do everything, but that they need a lot of help because the body does not follow. It is a very severe disease in all its forms. The most severe form is type 1, and people are so affected that normally they do not survive 2 years of age, and they cannot breathe independently or self-feed independently. Of course, they cannot even sit and not walk, of course. Then there is the intermediate form, which is the type 2. Children and adults with this form can sit but have never been able to walk, and they have also severe musculoskeletal deformities. They have respiratory complications, and in general, they are very, very dependent. Type 3, which is the so-called mild form, is the one in which patients can, at some stage in life, walk, but they lose disability, sometimes within weeks or months. Sometimes it takes some years, but they always lose disability. So just to stress the fact that this is what we call mild form. I have never heard about a spinal cord injury being called like a mild situation, and that would be comparable to the most fortunate of people living with SMA. And I'm saying this because I will bring it later. As was already mentioned, since 2017, the world changed for us, and from talking about science fiction, now we have science in our side, and there are 3 approved therapies, one of them being Zolgensma, that has been already mentioned several times in this session. I have to say about the 3 therapies is that all 3 of them treat the underlying cause of the disease, and that they have all demonstrated greater efficacy the sooner you treat, but they have not demonstrated more efficacy on a certain population group or more safety issues on a certain group. It is a spectrum disease. The mutation, it's always the same. It's a lack of a gene and a backup gene that doesn't work properly. And therefore, as I say, there is no differences in the way the therapies act across the spectrum of the disease. So, Zolgensma 2020, and the EMA recommends its approval. The label that came out came as a very big surprise to all of us. We did not expect such a label. First of all, because it had no age, no weight restrictions. And secondly, because it was quite broad. It did have some limits. It was recommended for type 1 SMA, so the most severe form, or for people with up to 3 backup gene copies, which is the biomarker we have. It's not great, it's not perfect, but especially if you're looking at newborn screenings and you want, diagnosis for newborns, and you want to try to print anything better. But as I say, it's not a perfect biomarker. So what were the reactions to this EMA label? First, there was a quick doctor's reaction. They expressed their concern about whether the gene therapy would be adequately used in a safe way by doctors that were not that expert on SMA, because it is a rare disease, it is not that well known. So they published a consensus statement with guidelines for treating doctors. The consensus was published, and shortly after, us, patient representatives, we reacted to the doctor's reaction by publishing our own letter, in which what we basically said was, yes, we are generally in agreement with doctors, but please, be careful not to be using these treating guidelines as inclusion-exclusion criteria for the reimbursement negotiations, which then happened, as we will see. The reaction of the company was setting a $2 million price, 2.26 euro, as million euros, sorry, as Martina has mentioned in Germany, per dose, and establishing what they call the Global Managed Access Program, which was in fact a lottery, that they were giving 100 free doses per year in countries that had not yet had reimbursement agreements, and both the approval, the price, and the lottery took a toll on the community, of course. There was also, I have to say, a big hype around this Zolgensma, the gene therapy. As I was saying before, none of the 3 treatments for SMA have demonstrated a superior efficacy, but this one is a once in a lifetime, so obviously it is very attractive. So all of this led to a very strong emotional burden on the families. Families started fundraising across the world. I think you had your own examples here in Portugal. Families started relocating, and they still are, as also we have heard about recent news here. So all of this reverted in a pressure on the reimbursement negotiations, which did not make life easy for HTAs and reimbursement bodies. It was a not great situation. And so what did HTAs and reimbursement bodies do? They were very creative, I have to say, and they were not homogeneous in their outcomes, but generally they were all quite restrictive. They were not in accordance with the EMA label. So some limitations were related to age. Some as narrow as 6 weeks for newborns, for instance, in Spain. Others took the ages for the inclusion criteria in the clinical trials. Some took the FDA label, which was 2 years. Some limited by weight for safety reasons. It's true that there was very little evidence on the safety for bigger or heavier patients. And these gene therapy, I am not a physician, but apparently it does carry a very large amount of virus. So there were reasonable safety concerns. Other HTAs decided to exclude the milder forms, as I was saying. That would be the ones that are never able to walk and that have to sleep with ones that maybe walked until the age of 3. And this, because of the greater benefit for type 1s, well, it is a progressive disease. So everyone agrees that stopping a progressive disease and being able to preserve some autonomy and some independence is a great added value. But it is difficult to find a way to fund that or to put a price to that. That's different to the price for type 1s that are surviving and walking. Anyway, so also limitations with this imperfect biomarker that would leave some very severe cases out. Some schemes, some countries try to use payment per results with different installments, but as a previous speaker was mentioning, the difficulties come on which milestones are you going to be able to establish, and also what happens if with time the therapy starts losing effect. So not even this is like the perfect solution. And some countries also used name programs. So a big variety, as you can see. And unfortunately, this is the access map in Europe, being green where there is a good access, and the rest is still there is room for improvement. So just some ideas on how the situation we as patients think that could be improved. We definitely think that HTAs and reimbursement bodies need more support from all of the actors involved, especially regulators, expert clinicians, and also patient representatives who we are very knowledgeable on our disease, on our unmet needs, and on the benefits that therapies can bring to us. Also, it is important that the regulator acknowledges what is happening downstream with the decision that we make. I think that increasing transparency on the pricing and reimbursement negotiations would be very interesting. This is particularly hurting because for Zolgensma, all the basic research was funded by families. So we are really behind the patent, and then we can't access to the therapy. And with the reimbursement negotiations, when you hear from the outside what has happened, you really are saying, if I had been there, I would have had lots of arguments to put on top of a table for this or that, or anyway. It would be very important to develop new methods to reimburse ATMPs, and this should be a priority. I mean, science is there. When my children were diagnosed, everyone said, like, well, don't worry, science is progressing so much. They didn't know what to say. But then science progressed, and now us human beings are not able to find a way to fund this science and make this science accessible to patients. real-world data. I think it's a huge issue. It should be collected also for reimbursement decision-making. And therefore, it's not only about funding, but it is about supporting registry owners and engaging in early conversations with them. And then finally, really to set as a priority for everyone that patients access in a timely and equitable way. And by equitable, I mean in the positive way. If no one accesses, it's also very equitable, but it's not ideal. And I am just finalizing with some patients that did not access to gene therapy. These were milder forms, so no access in Spain. Mild form, she has a gastrostomy. Mild form. Too severe, she's not, how do you say? Her condition is comparable to as if she was dead, so she could not access either. And thank you very much.
So I must say that I'm very proud for the issues we brought to this session. They are so rich that I can't make a summary of it, but there was things that I would like to highlight and then give the word to the panel just to see, to find out if you want to contribute more. So it's clear that we have a different perspective when we are from the side of CAT and CHMP and approving or recommending for the European Commission an approval of a medicinal product for a rare disease, ATMPs in particular. From our side, we try, with the data that is submitted to us, to make the best of it, to reach the conclusion that there is a positive benefit risk with that data. More and more, and this is not limited to ATMPs, we have to say, even to small molecules, to monoclonal, more and more we are facing the less comprehensiveness of the data and the pressure to quickly, early, the approve or recommend for approval a product with the data that is still to be, clinical trials that are still ongoing, with limited size. And this is, like I said, this is not for ATMPs only. And the way we think is that, and that it was covered by many of the speakers in the first session as well, is that there is the need for all the effort that we make previously on the marketing authorization application assessment towards the patient access, a more close dialogue. So we all have to dialogue. And that has been done, an effort on the scientific advice for the parallel scientific advices, and also dialogue not only in Europe, but with other regulatory authorities as well, because the companies, these ATMPs in particular, are global, and some authorities ask different things that we are not aligned with all the requirements from other authorities and the vice versa. So the dialogue is important between the regulatory assessors for the HTAs and for patients. So I think this would be a good way to start. Also, because we are approving products that are, like I said, lack comprehensiveness of the data, I must say that we only approve them because the data that is present to us shows that there is a benefit risk. But then we have the other issues, that is the post-approval commitments. And when we are talking about conditional marketing authorization and marketing authorization under exceptional circumstances, there are the specific obligations that we agree with the companies. But when we do agree with the companies, we have to be sure that they are feasible. Because in the end, we gave the marketing authorization, and then in some years after, it was not feasible to set the obligations that we set. So also in that, we have to be careful and to be real on what we ask for.
And in this case, also there is some the mentioning of getting real world. That is probably the best solution in the future for all of these our uncertainties at our level before the marketing authorization, at the level of HTAs. And for that, there is a lot of things going on in Europe right now on trying to get reliable, unbiased, as much as possible, real world to support the post-approval, but more and more to support also the clinical trials that are limited in size and without comparators, without active comparators. So this is, when we talk about ATMPs, we have to be open-minded. They are special medicinal products, and we have to be open-minded in all the life cycles of the products. So these are my reflections, and I would like to hear from you that something that I said makes sense and you want to contribute a little bit more before I give the words to the audience. Martina?
To start, if you can hear me, yeah. Well, maybe three things. Data, clinical evidence at marketing authorization. I think that, yes, we're challenged a lot and with clinical data and marketing authorization. And I think the robustness of clinical data should not be in question. And I think that, yes, giving a clear message to the developers that a limited patient number with a limited follow-up in a non-randomized trial may not be sufficient, and that they should really make a strong effort, which is, of course, always linked to feasibility, to provide robust clinical evidence. And maybe more randomized controlled trials can be done. But it's of course always a case-by-case scenario. What can be done, like for a product where you have no comparator, there is no treatment available, then yes, you need data to contextualize, and that data is then based on the natural course of disease, and then you need really good real-world data. So, but I think that robust evidence at marketing authorization to enable our benefit risk assessment cannot be emphasized enough. The second thing is real-world data, and we've really, with the ATMPs, made very mixed experience with the quality of real-world data, both in the pre-authorization period, when we needed to contextualize the single-arm trial, but even more in the post-authorization period, where even a registry that had been certified by EMA to be suitable for a collection of real-world data proved 3 years after it had collected and provided us the data not to be suitable at all. So real-world data will not, and that's my prediction, replace clinical trial data. But when we realize, and in that case it was so, that the US-based registry provided much better data than the EU-based registry, then there's something wrong. And so this to navigate the European system of registries, to make a registry, let's say, for spinal muscular atrophy capable of including a gene therapy takes 2 years or 3 years, and depends, as was said, is the registry supported? Can it be supported? We have sometimes situations where you have a registry holder that is asked by the HTA to be the registry supporting the HTA real-world data, and the registry holder says, I'm not allowed to accept money from the industry. So it's a catch-22. It's the support, the identification, the interaction of European registries from the different countries, their way, the possibility for them to capture data of a gene therapy is something that we really need to work on. And there is a lot of talk about Darwin, but if you look at it, Darwin and the European Health data space, it's taking some years until it's working, and in between what we are doing in between. And then the third point that I want to make, you talk about better interaction between regulators and HTAs. Yes, and that's going to be fostered now by the HTA regulation and by parallel advice. But I think we're talking a lot about better interaction and planning of post-authorization studies. Yes, that is an important point. But we also have to recognize there are different tasks. And an HTA, the one that I know, is always saying, unless we have randomized controlled trials, you have a problem. And we say we don't need a randomized controlled trial. So we do have different different tasks, and the question is how much we can proceed by talking. I think it needs more than talking and more than communication. It's also about exchange of information so that HTAs have the scientific knowledge, the difference between an AAV and another type of gene therapy, a lot of things, but I think we also recognize the different tasks that we have, and we may often not have a randomized controlled trial, and the HTA is asking for a randomized controlled trial. So some critical points also from my side, what can be done, and that something needs to be done urgently, and that's about the European registry landscape.
Claudia, do you want to add something or not? Maybe not.
It's just a short comment. You said that we have to have an open mind when dealing with HMPs, and I think at the HTA assessment we have an open mind when dealing with HMPs. Otherwise, we will not have approved any of them. But we cannot have an open wallet, and the price of the medicines are probably, and Nancy mentioned that most of the funding was coming from the families with spinal muscular atrophy. So why are we paying such a high value? Probably more transparency on that price, and a more fair price for those medicines, because HTMPs does not mean that it has to be such a high revenue for the companies.
And Vincent, Philippe, and Francis, do you want to?
Yeah, I would just very quickly build on what Martina was saying. I do agree that we have to start by dialogue, but then I agree that, we have to establish the adequate whatever system, task force, exchange, and I'm sure we are able to develop new methodologies, and we can change the paradigm and ensure that ATMPs can be financed in a reasonable way.
You want to contribute,
Francisco? I would just like to say that this has been very interesting, because from a physician point of view, all I want is that the medicine arrive fast, because we read the clinical trials, then we read that the FDA approved, then the EMA approves, and from that, so that we can use them in clinical practice, it's always a matter of years. And the patients know about it, we know about it, and it's in diseases that we don't have that much to offer, is always frustrating. I understand everything you're saying, and I couldn't agree more. We have to be sure that the right therapies are approved, but it's very frustrating for us as physicians to wait for the decisions to take place. And
I will change a little bit the question to Francisco, because we are talking about the price of the ATMPs. And you mentioned something very nice in your slide, that is you produce some sustainable products. So why do you think ATMPs reach such a high price? From the perspective of a developer who is starting, but still with the notion that it's difficult.
It is, and I'm comfortable talking. I'm not from pharma, but we all know that developing ATMP takes a lot of years, even before entering clinical trials. It requires significant investment and a very high risk in this type of therapies, mainly because the ATMPs that we see reaching the market now, starting a lot of years ago, when both the regulatory framework was not clear yet, the analytical methods tools that were not adapted, even the GMP for ATMPs is quite recent when we think about it. And I remember discussing that with Margarida before we had those GMP. How can we adapt our process, manufacturing process to these guidelines? And it was tricky. So it's always something that, and even then the clinical trials also require significant investment. So, and the companies in the end have to have some type of return. So it's important to find a fair price that make this available to patients, but still, if we get there, the companies won't be able to invest next time around, so to produce new ATMPs. In our case, we are focused on that, and it's easier for us because we are dealing with ATMPs for small populations and where the manufacturing process is relatively easier outside, most of the times, in our case, outside the intellectual property network, and that also makes it easier for us. Simple, we are not a pharma, we do not intend to take that full way only by ourselves. So, and it's also important to mention that, and we see this a lot, clinicians and patients know that there are therapies in development, but they are not, patients have access. But Portugal is not really famous for having a high number of clinical trials. We are improving that a lot. But that's where we come out and we understand that, talking to clinicians, that hospital exemption could be a way for them to provide patients with access to therapies and at the same time gain knowledge outside the clinical trial framework, which is much more strict than the hospital exemption, to be comfortable in using this type of products and understand, for instance, what type of population might benefit most from it.
Thank you. You are mentioning two words that I'm sure we would have a lot of discussion, which is hospital exemption, but we are late already. And I would like to give the chance of the audience to ask questions to these wonderful speakers. So I invite questions from the audience. I hope. No? It's too late. It's too late now? Well, but so I would like to, so then I will have to ask something on hospital exemption. I'm sorry. Because, it is it's a difficult one, but it's something that connects what we are talking, which is patient access to medicine approvals. And from the experiences we got, don't you believe, all of you, that, or what do you believe what is the place for hospital exemption in the drug and in the development of an ATMP, not as the end solution? So the way we see it, we should see it as a way to get to marketing authorization, to get to patient access. That's the only, personally, I see hospital exemption. That we end up with this not consensual issue. Martina.
Well, it's the German position as well, that in principle it could be a way to support a clinical trial and enter then a clinical trial. It's difficult for us to see it as a replacement for a marketing authorization. And I mean, even for CAR T cells, I just mentioned the example of Spain that everybody was talking about, but they had very limited number of patients and most were in clinical trials. And we have in Germany also the question whether a CAR T cell could be in hospital exemption. And then what you would need as a basis for that is that with that, let's say, CD19 targeting CAR T, you have that first in a clinical trial. And why would we like to have that first in a clinical trial? Because a CD19 targeting CAR T is not a CD19 targeting CAR T. You know that they have different co-stimulatory domains, they have different fragments, different affinity to the tumor cell. So you have in the structure, in the cell selection that you have for a CAR T, CD4, CD8 ratio, whatever type of cells you have there, you have differences. And it's a very delicate balance with all these differences that you have that need to be well characterized to really make a CD19 CAR T an effective and safe CD19 CAR T. So the concept that you have that the one CD19 CAR T is the same as another one without having proof of at least a clinical trial is certainly the wrong concept. It needs to be based on science and it needs to be based on clinical data. So I have a very, very strong position there. Based on quite a lot of experience that we've had and also experience on looking at the data. So, and the other thing, hospital exemption, I think you need, with all the risks that we have seen, with also the cut paper on unproven cell therapies that we have published, you need, as a country and as an Infarmed, a good signed database to give a hospital exemption to a product. And I'm sorry to say that, but none of the mesenchymal stem cells so far have made it to approval. And why have they not made it to approval? We have seen a lot of randomized control trials with MSCs that have not been able to show safety and efficacy. So for me, it's a call to be very critical about the data, to look at the data very carefully, and to build on data from a clinical trial before an agency, and that's really my very strong opinion, should give a hospital exemption to a product.
Okay.
No, that's not maybe making friends, but I think that's it's a must. You need a mechanism of, you need a proof of principle, a non-clinical proof of principle. I mean, for a keratinocyte layer used for burns, it's something that, I mean, you can easily generate proof of principle. But for claiming that a mesenchymal stem cell is efficacious in whatever indication, you need proof of principle and you need clinical data. Because otherwise you put the risk on the patient for giving him an unproven or an unsafe or even a non-effective treatment. Personal opinion.
Okay. Yeah, I knew. I wanted to hear more, Margarida. That's why.
Okay, now we're getting a discussion here. Isn't that nice?
Yes, Margarida.
Yeah, well, I think it's very interesting to require clinical data, but the only way you get clinical data is to have a hospital exemption or a clinical trial.
A trial, yeah, you need a trial.
The problem of hospital exemption is that you don't have to do a clinical trial, and there's no mechanism to mandate that the hospital exemption is based on clinical trials. So the two key ingredients of the hospital exemption has been the number of patients exposed, and I think one important aspect that are in the Portuguese legislation, because I did it, is to have an annual report with full information of every patient that has to be assessed yearly. And so the limited number and the limited exposure and the results have to be assessed, and you can withdraw the authorization. Yeah. Because that's not necessarily a clinical trial. As you know, a clinical trial involves a totally different organization that is not centered in a hospital capability of generating cells and administering cells when you don't have a substantial manipulation.
Yeah. Okay, but have you? I'm sorry. Just one word. When you have when you say first do a clinical trial, and then show that there is some safety and efficacy, and then bring that product to the hospital exemption, and then come back every year with the data, I think that's fine. Have you ever looked at clinical data that come from real-world data that are you will have a big problem if you say hospital exemption, you don't need a clinical trial before, you just follow the patient and report yearly. And if these are real-world data, it will be non-interpretable.
Yeah, well.
Okay, but that's...
We have to finish, but even there, I think what I started this moderation is the dialogue, and so the people talk, and even the collection of data, the limited data during any use of the product, in order to be useful later in the regulatory procedures, is very important. And so that's why also authorities are making efforts to teach academics how to proceed their development, so that in the end patients have truly access of our research. And we have to finish. I thank you so much for your presence here. I sincerely think it was an extremely good session, and we will end up with our president of the Infarmed, Martina. You stay here, and we invited also Miguel to come here. Thank you all.
For the closing session, I ask Fatima Ventura and Martina to stay, to remain on stage, and I call the President of Infarmed and Erika Viegas from the Management Board of Infarmed, and also Professor Miguel Forte to the stage. Thank you.
No, I will be very short. I wanted to have here together with me to do this closing session. So we have the two moderators, Miguel Forte and Fatima Ventura. I mean, Miguel Forte was a bit more than a moderator because he was also very much involved in the organization, in the setting up of this conference. So it was really a partnership that we established between us and the society that Miguel is now about to preside as president-elect. So this was how we managed to put in place this group. And of course, I wanted also to have the two keynote speakers. Bruce Levine, unfortunately, as you know now, the real reasons why he was not able to come. So he's not with us, but I have the pleasure to have Martina Schussler-Lenz, and I'm very pleased to have here also another of our partner agencies, the Paul Ehrlich Institute in Germany, which is a very prominent agency, I would say, in our networking system. So I wanted to have here the keynote speakers because they were like representing all the speakers that are here. And of course, I want to share also with my colleague Erika Viegas from the executive board, because the other colleague, we are three members, is in Porto in another meeting, Carlos Alves, so he's not here with us. So my word is I really had moments where I was being pushed to leave the room, but I managed to stay the entire morning, and I think I did very well because we had a very rich morning with the two sessions. This was really very important also because they are illustrating the issues that we need to really discuss. And I think we have two contexts now that we have to be very careful. One is the implementation of the HTA regulation, and here, as Martin was also highlighting, but all the other speakers, we need really to work together. So perhaps these joint consultations that are in the HTA regulations, but we have already very well established in the side of the regulatory system, we have really to do it together and start doing it. There is a pilot now, which is actually run by another German agency, IQWIG, with the GBA. So this will be certainly very important in the setting up of the HTA system. But of course, also this is very relevant to the upcoming discussions that Margarida was also putting the flag on the discussion of the legislation, because this will be very important discussion. So I think it was really a key moment on this number of meetings that we have been organizing in the context of the 30 years of Infarmed. So it was really important. I want to say strongly on behalf of Infarmed, a big thank you to all the speakers, to all the participants, of course to all my colleagues that were behind the organization, also to the number. I noticed that there were dozens of people following the session online, so they were also following the morning online. So thank you all for being here, and I think you have been using well your morning of the last day of the week. So thank you very much and have a good weekend. Thank you. Thank you.
With the permission of the presidents of Infarmed, we close this event celebrating 30 years of Infarmed. Thank you for your presence and wishing you all a continued good afternoon.